just I want to give you one instruction that before I want to start my lecture, that I'm using my department Wi-Fi, and then sometimes occasionally the Wi-Fi goes off, and then in that case, please inform me, because in the presentation mode I cannot make it out whether the Wi-Fi is on or off. So that is my kind request. Thank you. So the topic of my lecture that I have chosen is fish and gish, uh, the molecular cytogenetic tools and their applications. Now, as a student of cytogenetics, probably you might be aware that there are conventional cytogenetics technique that we are using, like we are having the karyotype analysis, then we are having banding technique. So these techniques which we are conventionally using as the basic cytogenetic tools. But if you look into the problems of this, what we find that for example, if you want to do the karyotype analysis, always we need to focus on the metaphase stage. Okay, so that means we need to select a proper particular stage of cell division. Okay, and then most aberrations they are observed during meiosis cell division. So it is difficult to make out those changes. And then you know the next advancement that happened into the karyotype is the banding technique. So in the banding technique, also you need to use different dyes to develop the bands. Okay, but uh, you know um, these bands, they are restricted to the centromere or telomeric regions or heterochromatin regions. Okay, so if you want to see any changes in the euchromatin regions, then probably you may not be able to make it out. So these are some of the drawbacks with those with conventional cytogenetic techniques like karyotype analysis and banding technique. So as the science advanced, the molecular cytogenetic techniques they very evolved, and then a combination of the cytogenetic techniques and then molecular technique has given rise to the field of molecular cytogenetic techniques. Okay, so because of this, you know, combining these two powerful techniques, the powerful complement to the conventional cytogenetic analysis was made possible, especially if you want to analyze the aneuploidy, the structural rearrangements, like you are having chromosomal aberrations, deletion, duplication, inversion, translocation. Okay, then submicroscopic rearrangements, like if suppose there are micro deletions or duplications that have been taken place, then in subtelomeric regions, if some rearrangements that are taking place, you can easily detect that you know, mm, changes in the directly under the microscope using this molecular cytogenetic. So, so this is the slide that nicely explains how the combination of molecular technique and then cytological technique has been best used in a best manner. Now, even though we are the student of botany, we look forward for its best use for the betterment of human beings. So, this technique has also been widely used in medical science. Okay, so you are having one patient, and then suppose if that patient is suffering from any syndrome, then by using conventional technique like karyotype analysis, you need to construct the karyotype. Okay, you need to arrange those chromosomes pairwise in descending manner, and then you have to look for you have to look for the addition or deletion of the chromosomes by comparing with the standard human karyotype. Okay, so this is what the basic conventional cytogenetic technique involves. Now, on the other hand, you are having the different molecular markers that you can use, and then you can develop the banding pattern, and then like DNA fingerprinting pattern you can develop, and then on that basis also you can do the molecular biological analysis. Okay, but if you combine both these together, then the molecular cytogenetic analysis is possible, and then here if you can make out my cursor, you are seeing in the under the microscope field, you're observing some red color dots, and then you are observing some light blue color dots, fluorescing dots. So this is how directly under the microscope you can observe that. Now this indicates that probably at this position the chromosomes, they are extra or they are added, okay, or they are duplicated. So this is how you can detect the syndromes or addition or deletion of the chromosomes or chromosome aberrations that are taking place in the affected patients. So this is the overall flow chart of the molecular cytogenetics era. So now after the conventional karyotype and banding technique, in 1988, fish technique was discovered. A fish, it has nothing to do with our fish, what we are having as an animal in the water, but it is the abbreviation of fluorescent in situ hybridization. Then in 1992, the another technique, related technique, that is GISH was developed. So it is a genomic in situ hybridization. Then subsequently, those techniques were modified over the time, like 1994 reverse fish, 1996 spectral karyotyping M fish was developed, 1999 band analysis was developed, and then 2002 fiber fish. So primarily in situ labeling, and then 2002 microarrays. So these developments, they occurred in the field of molecular cytogenetics. So now, before going into the fish and gish, we will first see, that means fluorescent in situ hybridization and genomic in situ hybridization. We will see the what is meant by the in situ hybridization. Okay. Now, suppose as a student of botany, if suppose somebody comes and asks you that, okay, okay, you are saying that genes are there on the chromosomes, right? That is fine, and then you know. There are different tools, direct and indirect tools to locate that. So can you tell me, or can you show me under the microscope where, on which chromosome a gene of my interest is located? A particular gene, suppose X gene, is look. I want to see its location under directly under the chromosomes. So if such questions will be asked by your friends, some of the friends by the science students, okay, then you know what you will think about it. So through the conventional technique, what you will be able to do maximum that karyotype, you can able to construct the karyotype. Okay. Mm, and that is the maximum limitation you can have, that you cannot go beyond the constructing the karyotype, right? Then further, if you go one step more, and then you can develop the banding pattern in that using the different uh, chromosome specific dyes. But again, the question probably remains unanswered, that how to show under the microscope that where the gene of interest in which my friend is interested is located, right? On the other side, you're having different molecular techniques that are there. So using the molecular markers, okay, PCR based molecular markers are there, and then you can go up to the recent era or period of mm, whole genome sequencing and all that. Okay, so no doubt, they will develop good amount of information, and you can get good knowledge of the genome. But this question again cannot be probably answered using that, because those no doubt those techniques, molecular analysis techniques, they are highly advanced, and they can give lots of information. But the desire of the student to observe the gene under the microscope remains unanswered, okay, or unfulfilled, right? So if it is so, then how we can see the location of the gene on the chromosome, and that under directly under the compound microscope. So this question definitely can be answered by this particular you know, fluorescent in situ hybridization technique. Okay, so I hope you have got the point here that this is how the fluorescent in situ hybridization technique or genomic in situ hybridization techniques help you to locate or pinpoint the position of the genes on the chromosomes. Okay. Now, genes, I have given you one specific example because we are much more interested in genes. Okay, but besides genes, we are having different regions. We are having different euchromatic regions, different heterochromatic regions. We are having repetitive units. Okay, we are having telomeric ends. We are having centromeric DNA. So, if you want to specifically pinpoint these regions on the chromosomes, you can be able to do it easily, using this particular technique. So we'll go into the details of this technique. So before going to the actual fish and geese technique, we'll first clear our concept about the in situ hybridization. So you are familiar with the word in situ. Okay, so in situ, that means within the cellular environment. So this ish or in situ hybridization is a powerful method to localize nucleic acid sequences in vivo, that is in the tissue, cells, organelles, nuclei, or chromosomes by using appropriate probes. So we are taking the help of um, the markers, which we are calling it as probes, which will go and specifically bind to the site of our interest, and then those could be visualized using fluorescent markers or fluorescent dyes. Okay, so with each, the nucleic acids are localized in their original or proper place. So, what is the basic technique? Now, first you have to you know prepare the biological material of your interest. Okay. Then second thing about the probes. So, the probes is what is probe that we are going to see in detail in the following slides. So, basically, probe is a complementary sequence or a complementary region to the DNA or the chromosome region of the plant species of interest. Okay. And then these probes, they need to be labeled in order to make the binding of the probe and the complementary DNA visualized. Okay. And then these you need to denature these both probes and target nucleic acid, and then single-stranded probes gets hybridized to the region where it found sequences complementary to that, and then the hybridization is detected, and then hybridization is visualized. So these are the basic steps that are involved in the in situ hybridization. So now, what you can localize using this technique? Okay, so you can localize DNA sequences, RNA sequences, viral sequences using this technique. Now, as I was telling you, in DNA sequence, you are having repetitive regions, you are having unique regions, you are having you know you can consider a whole chromosome or part of the chromosome or whole genome. Okay, as such, in RNA sequence, you are having the RNAs. Suppose if you are specifically designing some experiments, you are having
Sir, unmute yourself, sir. Altaf, sir.
yes.
Yeah.
I was muted, got muted. I muted, I think. Can you hear me now?
Yeah, yeah, it is clear.
Yeah, yeah. So Now I was talking about the RNA sequences. So if suppose we want to study the pattern of gene expression under a experimental condition, like suppose you are given a salt treatment, okay, you are given drought stress, or if you are having biotic stresses, and under that, if you want to study the genes that are expressed, no, so these particular gene expression studies could also be possible by this in situ hybridization. And then viral sequences, the corona current case is the burning case which is going on. Whole world is suffering out of that. So such viral sequences, those could also be localized using this particular technique. Okay, the affected patients could be diagnosed with the help of the disease technique, the viral, the viral genome if it has got integrated into the human genome. Okay, so before going into the actual procedure, we will have some brief idea about the probe. So, what is probe? How we can define a probe? So, probe can be a DNA or RNA sequence which is labeled and used to probe or bind the target nucleic acid. Okay, probe is the DNA or RNA sequence which is labeled and used to bind to the target nucleic acid. Okay. So obviously, when it goes and binds to the nucleic acid, no, the probe has also got the complementary region, which is which complements with the or which is similar with the nucleic acid, where it goes and binds. Okay. Now, depending on the use, there are different categories of probes that are identified. So first is called as nucleic acid probes. So those can be either DNA or RNA probes. Now, based on the synthesis pattern of the probes, three different types of probes are identified. Like suppose if you are cloning your sequence of the DNA into a vector, so that and then you are synthesizing probe based on that. That is called as a cloned probe. Then synthetic probe, you can based on the already available information of the genome, you can design the probe, okay, with the help of the commercial companies. That could be called as a synthetic probe. Then another one that is through PCR technique, polymerase chain reaction, you probably may be aware of. So through this polymerase chain reaction, you can amplify the DNA of your interest, and based on that, if you can design the probe that could be called as PCR probe. Now, now, if you are aiming to study the complexities, okay, in the genome, like suppose if you are interested to study the repeated sequences, if you want to study the single copy regions in the genome, suppose if you want to study the whole chromosome or part of the chromosome or total genomic DNA, then a specific probes are designed, and those are called as the complexities of probes. Those probes obviously will be based on the complexities of the probe sequences. Okay. Now, as we have seen that the probe is complementary to the nucleic acid, so obviously it will go and bind to the nucleic acid, right? But we need to detect them, okay, that where it has gone and bind. Okay, so and for that purpose, okay, you need to label the probes. Okay, so I will give one example, common example, that you are having a group of 1,000 students in the class, and then suppose if you want to pinpoint a particular student in that particular group of 1,000 students, then how will you identify that? Okay, now you are knowing that in school, suppose there is a common uniform, and then under that common uniform, it is very difficult to identify a particular student. So suppose if you'll ask that particular student to dress differently, okay, suppose a different uniform or different color code uniform, if that student is wearing, and then suppose if it gets mixed into the crowd of 1,000 students, then easily you can pinpoint or identify that particular student. So this is how, why we could identify that student, because that has been labeled differently, right? So in a similar way, we can also label the probe so that we can find out where our probe has gone and bind to the to which region of the DNA. Okay, so in labeling processes, again there are two ways. One is called as enzymatic labeling. Here biotin and deoxygenin, these are the reagents that are helping for this. And then we are having the another type of labeling that is called as chemical based labeling, chemical labeling. So you can use acetylaminofluorine or mercury, for labeling the probes. Okay, then you know um sometimes these probes can also be labeled with radioactive isotopes. So such uh, probes, they are called as radioactively labeled probes. Okay, so you are knowing that the radioactive labels are the isotopes which emit beta particles and are detected by autoradiography. So you are having 35S or 32P or uh, 3H. So these are the radioactively labeled compounds which can be incorporated into the probes, and you can label that probe for this particular mm, labeling activity. Okay. Similarly, you cannot. I mean, if you are not interested to go for the radioactive compounds, then some other compounds they are also coming to our help, and so under that you can categorize them as the non-radioactive labels. Okay, so these are again falling under two categories of direct and indirect ish. Okay. So now you are having the direct and indirect ish that is explained over here in the slide. So in the direct in situ hybridization, the label is incorporated directly into the nucleic acid probe so that hybridization site could be visualized immediately after hybridization. Okay. The same example, that I have given the example of the student. Okay, the student has been dressed differently, and then because of that we can pinpoint or observe that particular student. Right. So this is a direct labeling. So what is done here, that the label is directly attached or incorporated into the nucleic acid probe. Okay, and it goes and binds to the site or complementary region, okay, or hybridization site, and then we can make it visualize under the microscope. And there is an indirect way of labeling, okay, of the probe. So here the label in the probe cannot be detected immediately after hybridization because it is indirect. So second molecule called as reporter molecule is required to detect the label in the probe. Okay. So I hope you are making out the difference. In indirect method, the label itself cannot tell that okay, I want and bind here, and it needs the help of another molecule which is called as a reporter molecule. Okay, so the reporter molecule binds with this particular label, okay, and it generates the signal, and by means of which the visualization is made possible. Okay, so this can be one or two steps. Now, now, because of the complications in using the radioactive labels, non-radioactive labels, they are widely used or preferred. And now, some of the examples of the non-radioactive labels are biotin, deoxygenin, acetylaminofluorine, and mercury. Okay. Then some of the reporter molecules that are used here. Now here avidin is used or streptavidin is used, to detect the biotins. Deoxygenin can be detected by anti-deoxygenin antibodies. Now this AF that is detected by anti-acetyl aminofluorine antibodies, and mercury is detected by the ligands having an immunogenic group which can bind to same antibody. So now, as we have seen in the indirect labeling, it generates the signal. Okay, so now different types of signals are also used. Okay, so one is the one signal is called as the fluorochrome signal, as the word indicates. It fluoresces. Okay. Because they get excited by the light of one wavelength and then emit the light of another wavelength, which is observed as a fluorescence of different colors. So that could be detected under fluorescent microscope. Then you are having the enzymes. Okay, so enzymes, they work by catalyzing the precipitation of the visual product at the hybridization site, and then the color reaction could also be detected under microscope. And then third category is of metals. So metals like colloidal gold, which is conjugated with antibodies and can be visualized with both light and electron microscope. So these are the three signal generating systems that are used. Now the enzymes that are used here, they are listed here like horseradish peroxidase. Now it uses the substrate DAB, diaminobenzidine, and it gives the precipitate, uh, color precipitate. So here this horseradish peroxidase generates a red color precipitate. Then alkaline phosphatase. Now here the BCIP is the substrate that is used, and it gives you the blue color precipitate that could visualize under microscope. Now you are having the different fluorochromes. Okay, now as I was telling you, they get excited under uh, particular wavelength, and then they emit the light that could be observed under fluorescent microscope. So you are having FITC fluorochrome. It gives a green fluorescence. You are having a tetramethyl rhodamine isothiocyanate, which gives you the red fluorescence. Texas red or sulforhodamine, it gives deep red, and then MCA, it gives you the blue color fluorescence. So now the technique has been advanced so much that you can do multiple labeling. Okay, and that suppose you are having in humans, you are having a 23 sets of chromosomes. Then each chromosome you can color separately, or each chromosome you can label separately using these chromosome specific probes. Okay, so in multiple labeling, more than one probe can be employed simultaneously on the target nucleic acid. Okay, and you can specifically label the different regions in the nucleic acid or different chromosomes. If now how this helps, this can be used to determine the relationship of the different sequences with respect to each other. Suppose if you are comparing two plant species together, one genus, two species, then obviously being they are having the common genus, they might be sharing some genomic regions in common, or some genomic regions may be different. So if you want to compare this, you can do multiple labeling. The second one is to identify different chromosomes simultaneously, as I was mentioning you. Different chromosomes, if you want to observe simultaneously, you can do this, and then you can identify different genomes simultaneously. So at chromosome level, at genome level, at sequence level, you can use this multiple labeling. Okay, and then accordingly types are defined. So sequential multiple labeling, simultaneous multiple labeling, which is indirect method, simultaneous multiple labeling by direct method. Okay. One example is shown here. So we have seen that biotin, is the molecule that is used here. So if you can make out my position of my cursor, okay. So at 3 o'clock position, okay, on the topmost diagram, single standard probe is there. Okay, and here you are seeing some rectangular color box, which is giving you the idea, or which is telling you that this particular probe is labeled. Okay, single stranded probe that is labeled. Now, in the beginning, we have seen that we need to denature the DNA as well as probe to make them single stranded. So here you are having the single stranded probe, which is labeled with this biotin molecule, and then you are having the single stranded DNA. Now they are mixed together. Okay, they are allowed to now uh, hybridize. The probe will go and bind to the complementary region. Okay, and then we have seen that biotin itself. Sorry, sorry, sir.
Sorry, sir. Not for you, sir. Sorry, sorry. You continue, sir.
Okay, yeah, yeah. So the probe will go and bind to the single stranded DNA to the at the complementary region. Now we have seen that biotin itself cannot be detected, okay, because it is an indirect way of labeling. So biotin needs a reporter molecule. So here the FITC is used, which conjugates with this particular biotin, and then it you know emits the green color fluorescence, and then those that could be detected under the fluorescent microscope. Okay, so you are seeing here, in the first step, the single stranded probe and the single stranded DNA, they are allowed to hybridize. Okay, the single stranded DNA, that is probe, uh, binds to the complementary region. Now here you are having the biotin molecule. So here we are taking you know the FITC as the reporter molecule. So in FITC there is a, I mean, there are different chemicals under FITC. Okay, so one is avidin, okay, that specifically goes and binds to the biotin. Okay, and when it binds to the biotin, under a particular wavelength, it generates the green fluorescence. We are seeing a green arrow here. It emits the green fluorescence that could be detected under the fluorescent microscope. So this is how the indirect uh, labeling of the probe works. Now, after this background of in situ hybridization, we'll enter into the actual fish and fish technique. I hope you are clear with this in situ hybridization. So, just before going, once again, I want to recap what we have discussed so far. That in situ hybridization, what are the raw material that we are using? That we are using the first thing that is probe. Now, probe is what is probe? Probe is the complementary region to the nucleic acid of our interest. Okay, so what is the function of the probe? That it goes and binds to the nucleic acid, okay, at the site of interest. Now, what is the site of interest? Site of interest can be, suppose you want to detect repetitive regions, you want to detect single copy regions, you want to detect telomeric ends, you want to detect centromeric regions, heterochromatic regions, euchromatic regions. So like this, depending on our interest, suppose if you want to specifically mark a chromosome, okay, or all the chromosomes. So this is how it can be the region of the interest. Okay, so your probe will go and bind. Now, no doubt probes, since they are complementary to each other, they will go and bind to the targeted nucleic acid. But we made to visualize this. So we are taking the help of your label molecules. So labeling can be done. We have seen that it can be direct labeling or indirect labeling. Okay, and then now before the hybridization, we make we need to make the nucleic acid as well as probe single stranded. So we need to denature it. Okay, and then when we will allow to hybridize it, the probe will go and bind to the complementary region. The signal molecule which is attached to the probe, it will give you the signal, and then accordingly you can detect where your probe has gone and bound to the region of your interest. So now in the in situ hybridization, these two techniques, they have been discussed. Okay, so one is called as fish fluorescent in situ hybridization, and then gish, that is genomic in situ hybridization. Okay, so in fluorescent in situ hybridization, a fluorescent molecule is deposited at the site of in situ hybridization. Okay, fluorescent molecule is deposited. So here the signal molecule is your fluorescent molecule, which is deposited at the site of hybridization. Now the site of hybridization can be a location of genes or DNA, okay, on the chromosomes. And then obviously being a fluorescent molecule, it will emit the fluorescence. Okay, that's why it is called as fluorescence in situ hybridization, or abbreviated as FISH. Okay, and in GISH technique or genomic in situ hybridization technique, what is done that total genomic DNA is used as a probe in hybridization experiments. Okay, so the total genomic DNA is specifically labeled, and then it is used to hybridize with the genome of the interest, and then it will go and bind to the complementary region, and it will give you the you know coloring reaction with the target nucleic acid, and that you can be able to detect it, using the devices. So we'll go one by one. First, we'll discuss about the fish technique. So this fish technique, it hybridizes a DNA nucleic acid probe to a target DNA sequence that contained within the cell nucleus. Okay, and then you can analyze a variety of specimens using fish. So the intact cells are attached to microscope slide using the standard cytogenetic methods, and then can go for that. Okay. Now, mm, no, another interesting part of this fish technique is that it can detect a single target. You can detect the two targets, or you can detect the multiple targets at a time. Okay. So for single target, you can use one color. For dual targets, you can use two colors, and then for multiple targets, you can use multicolors. Okay. So now, why this is important, especially multiple targets, because it allows to look at multiple genomic changes within a single cell without destruction of the cellular morphology. Okay, so this is what I was telling you, that suppose if I want to see the location of the gene on the chromosome directly under microscope, uh, that could be made possible without destructing the cellular morphology. Okay. So this is the beauty of this particular technique. So here, the probes again, probes used, the probes that are used here, they are the nucleic acids. That can be labeled with the marker, which allow identification and quantification. It will hybridize with another nucleic acid on the basis of the base complementarity. Okay, and then the site of binding can be identified, and not only identified, but we can quantify. So if suppose it goes and binds in a higher quantity, higher proportion, those could also be quantified, and accordingly you can see that whether a particular region is present in less amount or more amount. Okay. So the probe can be a part of DNA or RNA that is complementary to certain sequences of the target DNA. Okay. And then in case of here, suppose you are using in medical science, the DNA of the patient can serve as the target DNA. So if suppose you know we are using the different vectors like plasmids, fast DNA, plasmids, okay, or reacts, those could also be used as probes, or the PCR product. You can amplify certain segments of chromosomal DNA, and you can use that as a probe. Okay. Now, what are the steps that are involved in fish technique? So it is the same one what we discussed in the in situ hybridization. So first thing, that you need to denature the chromosomes. What is denaturing? Denaturing is the separation of the double strands into single strands. Now you must have learned it in molecular biology or genetics, that in order to separate out the chromosomal to DNA strands, you need to either heat them up, or you can treat it with alkali. Okay, any of the treatments could be followed, and that will denature or separate the double-stranded DNA into single-stranded DNA. Then your probe is also double-stranded, so that also you need to make it single-stranded. So again, you denature it, okay, make it single-stranded. Then allow the single-stranded probe to hybridize with the single-stranded target nucleic acid or chromosomes. Okay, and then you can go for fluorescent staining, and you can examine the slides or store in the dark. Okay, and go. So for example, here this is a flowchart that is been shown here, that you are having the metaphase cells or interface cells. Okay, you are having the DNA probe, so you allow it to hybridize, and then you need to remove the excess of the DNA probe through post hybridization washes, and then you can visualize it under a fluorescent microscope, and then you can do the analysis. Okay, so here some of the steps that are pictorially explained. How the whole procedure is carried out. So here you are having the specimen DNA, okay, which is single stranded. You are having the complementary probe, okay, and now you are seeing that this is the complementary region. So A G G C T A T. So this is the region on the your target nucleic acid, and this particular region is complementary region is present on the in the probe. Okay, so like A, here you are having the T which is complementary to A, you are having C which is complementary to G. So like this, you are having the complementary regions that helps to uh, go and bind through the formation of covalent bonds. Now another peculiarity of this probe is this, that you are seeing here a green color, you know, flower-like structure. So these are nothing but the your fluorochromes, okay, or fluorescent dyes that are bind to the probe. So here, in other words, the probe is fluorescently labeled. Okay, so this goes and binds to the specimen DNA at the complementary region. Okay, and then you can make it visualize. So this is another way of representation. Okay, so you are having the cells in interface. Now specifically, you have selected the metaphase stage here. Okay, now the in metaphase you are having the double stranded DNA. Okay, you didn't denatured it and you have made it single stranded. On the other hand, you are having the probe DNA. Now this particular probe DNA is labeled. You are seeing a red, red balls that are attached to the green color probe. Okay, so that means the probe is labeled with fluorescent molecule. Now you make it single stranded by denaturing it, and then you allow to hybridize it. Okay, now what happens that over the body of the chromosome where the complementary regions are there, it goes and binds. You are seeing the binding here. Okay, and this binding you can observe under the microscope field. Okay, now you are seeing here, the red dots, okay, on this chromosome. Okay. So like this, you can make it visualize. So this is how the fish technique works. This is another way of representation. Okay, you are having the probe DNA. Now you are labeled it with fluorescent dye. Then you are denatured it. You are having the this target nucleic acid. You are denatured it, made it single stranded. The single stranded probe will hybridize with the single stranded target nucleic acid, and then it goes and binds there. Under the microscope, you can observe this. This particular fluorescently labeled probe, it will fluoresce, and it will give. For example, here you are seeing red color fluorescence. Same yeah, that has been explained in another way. So you are having the tissue sample on your slide. Okay, now this is a magnified view. Now suppose you have specifically selected metaphase stage. So chromosomes are highly condensed here. Okay, and then these particular chromosomes, they are denatured. The fluorescently labeled fish, the probe is also allowed to denature and allowed to bind, and then it will go and it will bind and it will fluoresce. Okay, so this is how this is another way. I used many multiple diagrams so that the concept should be clear to you. So you are having the target DNA that is denatured, made single stranded. You are having the probe, labeled probe. You are seeing here a flower-like structure that are attached to the probe. So those are representing the labels. Okay, fluorescent labels. So the probe is also denatured, and then when you allow it to hybridize, the probe will go and bind to the complementary region. You know, and the probe has got the fluorescent signal or uh, label, that could be detected under microscope. So, nucleic acid hybridization is the formation of the duplex between two complementary sequences. Then intermolecular hybridization between two polynucleotide chains, which are complementary bases, can be done. So you can do the hybridization between DNA DNA. You can do the hybridization between DNA RNA or RNA and RNA. Okay, so any nucleic acid, any combination of nucleic acid you can use for hybridization purpose. Okay. Now you must have learned this in polymerase chain reaction. That is, annealing is the term that is used to describe the hybridization of two complementary molecules. Okay. So now, as the science advanced, the hybridization, could be atomized using the machines. So here, this is the machine. Okay, this is a machine that is called as hybrid company that manufacture this particular machine which hybridizes the probe and DNA target DNA together. Okay, so in a single go, it allows the probe and target DNA to be denatured. Okay, and then it allows the hybridization. So it is faster, easier, and a safer way of hybridization. So such automation is also made. Now, what types of fish probes we can use? So, different types of fish probes we can use based on the interest, our interest. For example, if you are interested in detecting the centromere region, they are called as centromere probes. You are seeing here a green color, so it goes and binds specifically to the centromeric region. So, if you are interested to detect the telomeric regions, okay, the telomere, that means the end regions, satellite regions, you can use a telomere specific probe and you can detect that. Then, as I was telling you, whole chromosome paint concept is also there, that you can paint the whole chromosome. We'll see in the following slides, and then that can also be detected. Or suppose if you are interested to detect a specific locus on the chromosome, that could also be possible through this particular field. So so depending on the interest, we can use the specific probes. So these are the actual real-time pictures that have been taken under microscope. Okay, so first picture, red one, that is a centromeric probe. It goes and binds to the centromere region. You are seeing it here, the yellow dots. Those are representing the centromeres in the chromosome. Okay. Now, locus-specific probe. Suppose if you're interested to know where my particular locus of a gene is present, so you can design the locus-specific probe and then you allow it to hybridize and observe under microscope, and then you are seeing here, dot-like structure. So they are representing, the locus. So what conclusion we can make? Suppose if these are the two different chromosomes, then your locus of interest is located on these two chromosomes, and it is absent on the rest of the chromosomes. On the rest of the chromosomes, you are not getting that particular, fluorescence. So this is how it is held. And then the whole chromosome painting, you can see here, that you can paint the whole chromosome, you know, uh, by single color. A single color means using the single fluorescent dye or multiple fluorescent dyes. So you are seeing here, if you can make out my cursor, okay, the yellow chromosomes are there. So which are giving the yellow fluorescence, that means they are the uh, homologous pairs. Okay, so green fluorescence they are giving, that means they are the homologous pair. Red fluorescence they are giving, that means they are the homologous pairs. So you can identify the homologous pairs, with the help of this whole chromosome painting technique. Or in this particular, the another insect, you are seeing that each chromosome, this is a human karyotype, human chromosome complement, and then each chromosome is differently labeled, and then uh, the homologous pair they are having the same colors. Okay, so like this, uh, you can use this particular fish technique very effectively. So telomeric probes, they have specificity for single human chromosome, or they contain locus estimated to be within 300 kb of the end of the chromosome. Then WCP chromosome, WCP means whole chromosome painting chromosomes. So whole chromosome painting probes. So the hybridized probe fluoresces with bright intensity along the length of the chromosome. So whole chromosome fluoresces here. Then CEP, that is the chromosome enumerator probes, which are specific to the centromere. Okay, so most are alpha and satellite 3 probes. Centromic regions are stained brighter, means they are rich in A and T bonds. Okay, then locus specific identifiers are there, which are called as LSI. Now, under that you are having deletion probes, you are having translocation probes, gene detection and localization probes, and gene amplification probes. Okay, then we have seen that we need to visualize these probes. So now we need to have a reporter molecule for that. So DNA probe is labeled with a colored fluorescent molecule. Okay, the fluorescent molecule remains attached to the DNA during the hybridization process, and then the molecule emits a particular color when viewed through fluorescent microscope that is equipped with appropriate filter set. Then we can visualize. So this is how, this is the fluorescent microscope you are having. The it is you can it has got the monitor attached, so you can visualize the cell directly on the monitor. So at 7 o'clock position you are having these filters here, different appropriate filters you can use. You are having the CCD camera which is placed on the top of the microscope here, and then you can take pictures of that using the CCD camera, and also you can have the images on the monitor. So this particular monitor, I mean, computer has got the fish analysis software that helps you to effectively analyze the your chromosomes. Okay. So multicolor fish. This is the next advancement in the fish technique. So it provides the colorized information relative to chromosome rearrangements, especially useful in specimens where chromosome preparations are less than optimal for standard cytogenetic binding analysis. So in standard cytogenetic binding analysis, suppose you are not able to get the optimal binding pattern or optimal preparations you are not getting, and then under this condition, you can use this multicolor fish. So this is a beautiful example picture that has been taken. Okay, so you can see here at 10 o'clock position. This is the how the homologous pairs they are nicely colored. Okay, now within the chromosome we are having multiple colors, right? And these multiple colors they are specific to the chromosomal regions. Okay, for example, here the end regions or telomeric regions they are white in color. Okay, so like this you can detect the heterochromatic regions, okay, different parts and so on. Okay. Now in the interface, the whole under the microscope, the whole cell observes like this. It is a multicolor picture, and then specifically, if you look into the metaphase chromosomes, you will find here, that each chromosome has colored separately and specifically. Okay, you can make a karyotype of that, and then what you are seeing here that the homologous pair they are colored with the same color, or they are fluorescing with the same color. Okay, and then you can detect for the changes, or for example in chromosome 2 here, this particular chromosome is red color. Okay, but its homologous partner has got some blue color at the centromeric region. So this indicates that this might be having some changes that might have occurred. Okay, and suppose if this is a karyotype of a patient, then you can correlate the that particular disease with this particular changes or abnormalities in the chromosomes. This is how you can selectively select and color the chromosomes. Okay. So here you are seeing a specific karyotype of a human. Okay, it is a female. Okay, you are seeing here a Y chromosome is absent. Two X chromosomes are there, and like this, you are having the color codes that are here. Okay, and then the chromosomes, all the 23 pairs of the chromosomes that are colored nicely. Okay, so now what you are seeing, for example, in chromosome 5 here, its homologous pair, you are seeing that you are having some violet color here. It is not a blue color, right? So that means some chromosomal changes might have occurred here, and that's why some different part from some another chromosome has come and attached here. And suppose if this particular karyotype is of one patient, then you can correlate accordingly, and then you can conclude that the disease or syndrome that the patient is having might be because of these rearrangements. Okay, so this is the slide which tells you the comparison between the conventional or traditional karyotype analysis and fish versus karyotyping. So that in karyotype you must have done this in practical lab. Okay, that must have obtained the metaphase stage, then you have constructed the karyotype karyogram. Okay, and then At the end, you are having the karyogram that is available. Okay. Now, suppose you are use the banding pattern, then you can also develop the ideogram. The ideograms are shown here. Okay, and then versus this, you are having or fish technique. Obviously, in order to construct the karyotype, you need minimum 7 to 10 days. Okay, first, you need to obtain the metaphase cell. Then, you need to have nicely separated chromosomes. Then you need to select the homologous pairs. You need to arrange them, okay, in a descending manner, and then you know that will give you the standard karyotype. Okay. But the beauty of this whipfish technique is that you can do the similar exercise within 24 hours. Okay, so you are having the cell, and then you use the specific probe that goes and binds. Okay, and then you can detect that directly under microscope, and this could be done within 24 hours. Okay, for example, here you know uh, here X chromosome at the bottom at 5 o'clock position, at 7 o'clock position, you are seeing X chromosome that is shown by green fluorescence, Y chromosome by red fluorescence, and 18 chromosome by aqua color. So so you are having you can correlate two cells. Okay, and then you can conclude that if some changes that might have taken place here. So in this diagram you are seeing single X chromosome. Okay, so that first thing that tells that this might be the cell of the man. Okay, and here you are getting you know 13 the chromosomes. I mean green dots on two chromosomes. Specifically, if you analyze that is present on the 13 chromosome, that means no, they are sharing some regions from the X chromosomes. Similarly, the red color dots you are seeing here, so they are sharing some chromosomes from the Y chromosome. So that means some rearrangements must have taken place. So such conclusions you can draw out of this. Okay. Now after this we'll come to the genomic in situ hybridization. uh, I hope I have some time to go ahead with.
Sir, if you can summarize, it will be better, sir, because other two speakers are waiting, sir. Please.
Okay, okay. So another is the gish. Now as I have told you, know the whole genome here is used as a probe, and then to use for hybridization purpose, and then uh, you can obtain the results. Okay. So this was for plant species. This was developed by Bennett and Hyslop Harrison. Okay. So same steps are there that are involved in in the fish analysis. So I'll just skip this. Okay. And then I'll come to the application part, okay, which is important. So now you can analyze the genome architecture through fish technique. Okay, so you can compare two species of a genus. Okay, or suppose wheat and rice. How much is the genome? What is our genome architecture? You can compare that species wise or genera wise. You can compare the repetitive DNA sequences. Okay, tandem repeats, telomeric sequences, centromeric sequences, microsatellites. Okay, you can characterize the genome. Here, in order to characterize the genome, you can go for gauge technique. Okay, you are seeing here. Um, in the picture, some of the chromosomes that have come from the ancestral species that you can detect. Okay. Then phylogenetic analysis you can do. Between the wild and cultivated plants. Then somatic variations in the tissue culture. You are facing with the somatic variation problem that could also be easily detected. So in this, you are seeing the red dot. That indicates that the plants obtained through tissue culture, they are having some variations. Then alien chromatin, you can detect. Alien means suppose if some chromosome or part of chromosome is added specifically into the cultivated species, that could also be localized. Okay, so the detection of the chromosome reanimates, that could also be done through this particular fish technique. So these chromosomal aberrations, you are knowing that inversion, duplications, okay, those could be. Okay. Then at the chromosomal organization at interface nuclei, you can see specifically you are seeing here the green dots, that are fluorescing at the telomeric regions. Okay, so this is how we can compare that. Then it is called as SNM fish, which specifically detect the centromeric regions in the chromosomes. Okay, then chromosome painting and diagnostic cytogenetics. So that is used for the diagnosing the syndromes. Okay. Mm, In the so this is how we are seeing that the telomeric regions they are fluorescing here. So centromeric regions they are rich in 80 bonds, so those could be visualized. Okay. Then telomeric regions could also visualize. Okay. So like this, you are having the telomeres that could be specifically targeted. So now there are some cases, but I think I will not discuss that because of the short of the time. I'll just keep this. So in medical field also this can be used. In transgenic animals also we can use this fish technique. For example, in poly this has been used specifically, and then can go for this. So and these are some of the ready references which you can refer to for your further reading and understanding the topic. So I hope you understood this, and then I
Thank you.
tried my level best to make you understand as much as possible. Okay, so thank you for your patient attendance. If there are any questions, if there is time for that, then definitely.
Sir, you stop sharing the screen and you switch on your camera, sir. I request Vasudeva sir, Ravikant sir, and Amrutavali ma'am, and others also to please switch on your videos so that we can have a snap. Sir may leave the meeting right now. Even Ravikant sir, you are there in meeting, please, Ravikant sir.
Hi Ravi,
Hello, Ravi. How are you?
unmute.
Oh,
Yeah, I
hello,
was.
hello. Yeah.
Yeah.
Hello,
Yeah,
Dr. Altaf. Also, long time no meeting.
yeah. I was not knowing that you are there.
Oh, okay. I didn't know, sir. Sir, what's this, sir? FBT or what, sir? What is that?
Huh?
Forest biology and tree improvement.
Oh, okay. You changed that. Okay. Nice meeting you also.
Me too, me
Dr.
too.
Ravi, we will have first Vasu sir's lecture and then followed by sir's lecture. Yours lecture will be there. Please
Okay.
excuse me.
Sure. No problem.
Thank you so much. Thank you, sir, Altaf sir, for such a wonderful presentation on hybridization. So you made the beautiful, I mean to say, slides of showing how the chromosomes can be mapped for several purposes, and it was really awesome lecture what we have seen today. So in future, we will be requesting you again for something like karyotyping. So your
Yes,
expertise in karyotyping, it seems, because the
Yeah.
slides and all says that you are the one who has used the latest technologies for karyotyping the chromosomes. Thank you so much, sir, for having joining for us. Thank you very much.
thank you, sir.
Now it
Thank
is
you
a
for
proud
giving me the
privilege
opportunity.
to introduce Vasu, sir. You can start sharing the screen.
Yeah.
Yeah.
It is proud privilege for the Department of Botany, Telangana University on virtual mode. We have Professor R. Vasudeva sir, Professor of Forest Biology, College of Forestry, Sirsi Campus, University of Agriculture Science, Harvard, Karnataka. His academic qualifications are he is PhD in plant breeding and genetics, University of Agricultural Sciences, Bangalore. Specialization is forest ecology, tree improvement, conservation genetics. Recipient of position and honors. Recipient of INSA Teacher Award 2018, constituted by the Indian National Science Academy, New Delhi. Recipient of Best Teacher Award, constituted by the Indian Council of Agricultural Research, New Delhi in 2016. Served as a member of Expert Appraisal Committee, Forestry, River Valley Projects in Ministry of Environment, Forest and Climate Change 2017-18. Served as expert member of Task Force on Conservation of Western Ghats, constituted by Government of Karnataka for 5 years, 2009 to 2014. Invited by the Government of Indonesia, Malaysia to train scientists for their country on the concept of Forest Gene Bank 2009. recognized as curriculum development expert in forestry by the United States aided program, and was invited to develop a curriculum for master's degree program in biodiversity and management in Nepal in 2016. Served as global team member on community biodiversity management that undertook studies in Brazil 2010. Worked as a member on the Research and Advisory Board of Institute of Forest Genetics and Tree Breeding Coimbatore 2012. Currently, external expert member for Board of Studies UG in Forestry Faculty, Tamil Nadu Agriculture University Coimbatore. Publications: he has published more than 120 papers and more than 12 book chapters, and his achievements are obtained a global patent, US and European patent on a new molecule on for an effective treatment against cancer from a tree species of Western Ghats. Over 10 major research projects from various sources have been completed, which largely focused on improving livelihood through innovation farming. Developed a field gene bank of over 100 aromatic pickle mango and about 35 local varieties of jackfruit, and distributed over 50,000 samplings of locally important fruit varieties such as aromatic wild mango, jack, kokum. Developed a forest gene bank for three endangered species, successfully recovered a critically endangered tree species from the verge of extinction. With these words of introduction, I would request Vasu sir to start his presentation. Over to Vasu sir.
Thank you, Dr. Ahmed, for this wonderful introduction. And first of all, let me wish all of you a very happy Wildlife Day. I, coming from a forest college, Sirsi, I greatly take this opportunity
Yeah,
to
so in that technique, let me
celebrate this
manage the
Wildlife
reach
Day
can
with this
amplify
talk.
and check, and that
I will
too
be
at
speaking
one
on
temperature.
forest vegetation
They
sampling,
find out that,
techniques
but there
and
are
protocols.
6 prima pairs,
Huh.
and using that they can find
Uh,
out the presence of one particular microbe. So
there is a background noise. I think I request all of you to kindly mute. Okay, thank you very much. Yeah. So let me introduce my college. We are situated right in this Western Ghats. As you see, here is the Sharavati Valley, which is the hotspot of biological diversity. Our college has produced 31 IFS officers, about 300 range forest officers. In fact, the whole of or most of the Karnataka Forest Department is today is manned by the alumni of our college and our college as well as College of Forestry, Ponampet. Today, we are now in the face of climate change, where monitoring the forest has become an important issue. Forest monitoring involves monitoring plant ecological changes, biodiversity, tree density, forest demography, and then monitoring the forest carbon stocks, and then how the forest products are produced and flown out of the forest, as well as monitoring fauna. In fact, there are several methods to monitor. You have plot method, transect method, point-centered quadrant, and quarter method. Today, what I will do is to look at only one method of permanent vegetation plot establishment. And these are some of the broad steps that we follow as we go about with the presentation. You will understand each of these steps. So, how do we study and monitor the forest vegetation? Uh, One of the most important aspect of forest vegetation, and there is no other method for monitoring the vegetation, is the permanent vegetation plots. And these are highly suitable for measuring, estimating, monitoring the ecological parameters. Let me tell you what exactly is a permanent vegetation plot. What you are seeing is a valley near my forest, near my town, about 20 kilometers from my town, Sirsi. And this is the Aganashini Valley. And then if you mark 1 hectare of the area here, and then monitor the all and each and every tree through meticulous marking and tagging, and then that becomes a, if you take a periodic and long-term monitor, if you do, that becomes a permanent vegetation plot. It's essentially a plot marked in the space, in the sense that in a forested ecosystem, the space is a constraint for the plants to grow. We have to now identify this particular plot in the space of an ecosystem within the forest. What exactly we mean by long term is it's simply that long term is defined as about at least 50 years with an initial planning horizon of around 10 years. Like whatever that you start doing today, you should be able to monitor it for at least about 10 more years. That we call it as a little long term. Why permanent vegetation plots are important is they are firstly suitable for long-term monitoring of the vegetation. As I said, there is no other technique other than the permanent vegetation plots which can be employed. And secondly, they can also be applied as the national carbon stock inventory. As you know, the carbon stock inventory is now becoming extremely important for every country. Just like we have a petroleum inventory or any other resource inventory, we should have a carbon stock inventory. And it's suitable for forests with different maturity types, density types, etc. And it's simple and cost effective when compared to many other techniques. And they're also very important to understand the structure and composition of forest, how exactly different forests look like, what are the different stratum that you have, how many, what's the kind of different life forms exist in each of these forest types. All this can be now looked into. We could also assess the growth rate of an individual tree. In fact, growth rate of individual tree is an extremely important data point for calculating the carbon stocks, and then how long a particular tree will survive in an environment. That's the longevity of the species is important. In a way, to understand the vegetation dynamics, over a period of 10, 15 years, the whole composition of a particular plant forest type might change because of various reasons. And then this is possible only through this. And we can also have some idea about what would be the impacts of human intervention, like the fire, and then if there are any space-based vegetation studies done, and these permanent plots will be the ground truthing sites. So hence, establishing a permanent vegetation plot is one of the most important tools for a botanist, for an ecologist, for a geneticist, for an atmospheric scientist, and today, as such, for a policymaker. But that's not very easy. This is one of the summit point in the Western Ghats. If you look at this forest, it looks very, very homogeneous. Everything is fine. So just make a small plot and it might tell you the whole story of it. But unfortunately, this is not the case. When you look at closely, you find a Western Ghat is a landscape with a lot of disturbed elements. For instance, what you see here is an undisturbed forest. This is a disturbed forest. You have a farmland, you have a grassland. So every inch of this particular land is important, both for the studies on the vegetation as well as for the carbon stock monitoring. What happens in the grassland may affect the total carbon stocks. What happens in a adjacent horticultural land might be important for drawing certain conclusions. Hence, it's very important that we should now consider every landscape element before we actually proceed. So, as we all know that it is almost impossible to measure every tree. Therefore, we need to take samples. The basic principle of taking a sample is to make sure that it's a complete representative of the population. Otherwise, the information obtained will be biased in some way, and then inferences drawn from the data are likely to be invalid. So, taking a right sample, hence having a right sampling approach is an important issue. The first and foremost thing before we monitor the forest and before we establish a permanent vegetation plot is to divide the entire forested area into sampling units. And next step is to decide which sample units to include in the survey according to the appropriate design. And then when you go into a specific area, we can have sampling designs such as simple random sampling, stratified random sampling, so on and so forth. So let me, this I'm taking the entire course of the talk will be as if this is a classroom for the students to follow. So that's exactly what Dr. Ahmed had told me, that it should be like whatever that you do for a PG class should be, you should be able to present it. So this is more like a protocol narration of the permanent vegetation plot establishment. The first step is the selection and stratification of the land use. Second is defining the project boundary. Third is sampling through plot laying. Within the plot laying, you have sub steps like marking the plots and subplots, making the measurements, etc. See, we know that a landscape can be now divided into smaller units, such as if you take any unit like whether it is in Uttar Kannada district or Kodagu or in Telangana, you have forests, you have plantations, and you have an agroforestry system. And within the forest, you might have different types of it: evergreen, semi-evergreen, and deciduous. And then in plantation, you may have teak plantation, acacia, or a mixed one. And you also have agroforestry systems like home gardens and beetle-nut garden. And if your aim is to look at the carbon stock of the entire landscape, it is very essential that we need to monitor each one of these landscape meticulously. So what you do in an evergreen forest should be replicated in all these units. Going one step further, all evergreen forest may not be as intact as we imagine it to be. There may be some amount of disturbance. So hence, it's also essential that we need to consider disturbances within these forest types. So essentially what we do is, let's say this is your Bedti River that's flowing through the Western Ghats. And then if you look at, you have this mushroom head like the forest, but unfortunately, not all these elements are like this. You have a lot of disturbances. You still, this is as per the law, this is still a forest, but you have a home, a township coming up here, and this has actually disturbed it. This could be one of the disturbed levels of, let's say, a deciduous forest. We need to consider the major forest types as well as the disturbance types. Now, this is the most important step in identifying before we establish a permanent vegetation plot. And essentially, stratification of the project area is nothing but disaggregation of the land area into homogeneous units. And then, of course, this is very essential to take the right sample of it, and so that the sampling error is reduced. How exactly we select these forest types is, we take the help from the space, we take the help from the GIS platforms to identify areas that are homogeneous, areas that are having a disturbance, areas that are having a thick vegetation and then disturbed vegetation, water body, etc. So using a remote sensing image, topo sheet, we can decide where exactly the plot has to be located, and then that has to be marked and appropriately on the map. Let's imagine I have decided to put my first plot in this evergreen forest patch, which is roughly around 300 hectares. Let's say this is the forest patch that I've decided. And then wherever you go in the Western Ghat, you have a village. Let's imagine that there's a small village. Now, my intention is to put 1 hectare permanent vegetation plot in this evergreen forest. Where do I locate my 1 hectare plot? Simply, what we can do is firstly found that the plot should be away from the edge of the forest. Suppose this is the forest edge, it should be away from the edge at least 3 times the canopy height. Suppose the canopy height is around 20 meters or 20 meters, then you must leave around 60 meters away from the edge, and then consider all those areas for plotting. This is basically to avoid the disturbance, laterite, the atypical vegetation that must have crept into the edges, in effect to eliminate the edge effect. So essentially do that. Now, let us say this 300 hectares can be now divided using GIS techniques into 300 plots like this on the computer, and then mark 1 to 300 like this. So this is my plot number 1, 2, 3, 4, 5 like that, and that is my 300th plot. Now, which 1 hectare of the plot we should consider, where exactly it should be located. Now, what we can do is we can now, after the stratification that we did, within this stratum, we can go for a restricted random selection of the plot. What we mean by that is you simply put cheat of 1 to 300 and then pick up a cheat and then look at the number. Suppose the number is somewhere here, do not select that, basically because it is in the edge. Let us say this is somewhere here, do not select, or any of these you can select. So I'm doing a randomization of the selection for the location of the plot with some restrictedness. So this is called as restricted randomized selection of the area where we should put our plot. Okay. Now, some tips like this. Do not select areas like this. Avoid locations which are having certain amount of disturbance because it may not be important. But suppose your project area is to look at the disturbance itself, then we should be able to consider this. So all plots should be randomly distributed in a chosen land. Avoid atypical vegetation, sharp ecological discontinuities. Avoid locations where there are road, rails running through, etc. Now, after identifying the location of our where to put the plot, what should be the plot size? Usually, as per the UNESCO's program of Man and Biosphere recommendation, you can either have 1 hectare plot or you can have 20 by 20 meter plot. 20 by 20 meter plots are essentially, standalone plots that can be, many of them can be put, and most of the permanent vegetation plots are of 1 hectare plot. So this decision has to be taken based on the resources and then how difficulty is laying a 1 hectare plot, etc. So both are actually recommended for long-term plot. So now let us look at how to establish 1 hectare permanent plot using it. Some of the national forest inventories also use some other method called cluster method of in a plot, where they put a permanent cluster of 9 clusters, and each plot here is a circular plot of either 10-meter radius or 7-meter radius, and each running in this kind of a condition. Of course, we are not following this. Only certain national forest inventories, permanent inventories are using this kind of a thing. Today, we will look at 1 hectare plot, a kind of a square plot that we are talking about. You might be wondering why 1 hectare plot? Why not 20 by 20? Why not half an hectare? In fact, 1 hectare in most of the tropical situations provide relatively large sample. It's quite a large sample. And it's also a reasonably good representative of the selected stand with respect to the patchiness, tree fall, species composition, etc. Suppose if you walk 1 hectare all through the 1 hectare of a forest in any spot in the Western Ghats, you will definitely find certain patchiness, you will definitely find 1 or 2 tree falls, etc. And then it will also avoid unintentional bias that might often creep in when you are trying to put only 20 by 20. So human mind is that if you want to put, especially if you go with the students, they often go to an area where there is less vegetation so that they will finish their work very quickly. But when you put an 1 hectare plot, this includes thick, thin, and mediumly disturbed kind of plots. That's why 1 hectare plots are necessary. So essentially 1 hectare plots are divided into subplots, especially the subplots are of the 20 by 20. So suppose if you take 100 meters by 100 meters, that constitutes 1 hectare. And then within these 1 hectare plot, you might have when you split this into subplots of 20 by 20, you have 25 subplots of 20 by 20 dimension. So preparing the plot is extremely simple. All that you require is rope, tape, peg, and then activity involves very physical activity of marking the corners using a peg and then recording GPS readings, etc. So first thing about it is that you need to locate the plot wherever you are marking your first plot. The starting point has to be located using GPS, and any other permanent visible landmark on the field has to be now looked into. And then second is mark the corners of the subplots on the ground using either the aluminum pegs or the very solid rock pegs like this. And then every sub corner, the GPS readings can be now taken so that you will not miss out. See, suppose even if it is 1 hectare plot, if you are in the middle of the plot, you will be easily confused regarding where exactly you are. So it's essential that every corner has to be marked with respect to location of which subplot you are in. Suppose you are here, you should be able to see that this is subplot number 13, and down below is subplot number 9, so on and so forth. So such markings are done like that. So this is how a marked 1 hectare plot would look like with the peg, okay, like this. And for the vegetation sampling, we go for nested subplot. What do we mean by nested plot is, suppose you ask your students to count all the herbs in a 20 by 20 plot, it will take at least 3 days to put that. So there are so many herbs that are there present, it's almost impossible to count within the given period. So for all the tree component, I consider 20 by 20 as my sampling unit. For the shrubs that are present, I consider a smaller plot of 10 by 10. And for the herbs, I consider 2 1 by 1 plot of 1 meter by 1 meter. So this would be my subplots. Okay. When we enter into a plot, what we do is we identify all the individuals of the species. Hold on a second. Sorry for the disturbance. So when you enter into a plot, we will identify all the individuals in that plot to the species level, tag every individual with a unique number, and then make measurements like the diameter, height, and other conditions. And then some observations are made in the shrubs and herbs separately. So let us imagine that we have already inside a plot, and then we have to identify a tree. Then what exactly is a tree? Tree generally is defined as that individual which is more than 10 centimeters, which is having a girth, and then at the breast height, that means 1.3 meters from the ground. And then sometimes what happens is that in some of the drier part, even some tree species which are having less than 10 centimeter girth would have flowered and fruited. Then that is still to be considered as a tree species. That should be included in the tree layer. So based on the local conditions, you can preset this condition, whether it is equivalent to or more than 10 centimeters or equivalent to more than 5 centimeters. Based on that, you need to make such corrections. Now, within a plot, the parameters considered are as many. And then what exactly is done to establish 1 hectare plot is, let us imagine that this is your 100 meter by 100 meter plot. Firstly is to select or identify the corner of your plot. This has to be done using a theodolite or a forest vegetation plotting. Okay, so you need to use certain methods of theodolite and then make these cornerstones most important. So A, B, C, D first one. So mark, select the area for sampling, mark and lay the 1 hectare plot. And then next is that within each of these 1 hectare, we have to make strips of 5 strips, each having a width of 20 meters and a length of 100 meters. So divide this entire 1 hectare plot into strips of 20 meters of width and then 100 meters of length. And then each of them we take, so we go to one strip here and then start making the subplots of with 20 meter by 20 meter grid. So essentially, first mark these cornerstones and then make into 5 strips. And then within each strip, you make 5 subplots and you will have 25 subplots present. So once you have the tree enumeration, you can start with corner A and then confirm with tree definition, identify the tree species, mark at 1.3 level and measure the diameter. Either you can measure the girth or you can measure the diameter based on whatever you want to do. So usually a marking is done of the starting point. And for all permanent vegetation plots, marking with an aluminum tag is an essential, basically to re-identify, re-monitor, and then come back, and after 5 years, you have to take the girth of the same individual again. And then if you want to make what would be the girth that is it has achieved, we have to now do that. So affix an embossed metal tag to each of the tree. And if the tree is not going to, it's small enough, is less than 10 centimeters, you can tie it with an aluminum rope. So that's the kind of it. So measure the girth, and then how to measure the girth, I will let you know about it. And we also have to look at what is the level of disturbance that each tree may have. So in this particular tree, there is a heavy lopping that has been done. And then here you have, this is quite a top end is dead. And then you have to make the measurement of the girth. And then the girth measurement usually done in the following way. So suppose you have a tree and there is no branching before 1.3 meters above that, you can make it. And if there are 2 branches, you have to measure both of them. And then what you should do is, if this is a leaning tree, then measure the tape here and take the diameter there. Do not take that as your measurement. And if a tree is having a stilt root or some other root like that, and then you have to take above that 1.3. And then if it's a fallen tree, if it is having a exactly at 1.3, what to do. And then if you are in a slope, and then always take from the upper end of the slope 1.3. Don't take it from here because you may underestimate. And if it is a leaning tree, take along the length of it, of each of the branch, and then take it. So what I would do is to also circulate a small ready reckoner for you, how to do these measurements. I will supply to Dr. Ahmed so that it can be recirculated. And what we can also do is, or what we generally do when we enter into a plot, is to also look at what's happening to the tree, whether it is standing, it is a dead top, if it is broken, if it is leaning. So we give different, in acronyms. AS means standing, AD means dead top, AB means broken, so on and so forth. So we do height measurement. Height measurement is done using simple graduated scale. If your friend's height is, let us say, 6 feet, and he measures half an inch, then what would be the total height of it? So that's a very simple method. Or we can estimate, for instance, this particular palm can go up to 70 feet, or the small tree can go up to 10 feet. So if this electric pole is around 25 feet, so now based on our experience, we can even categorize. And then this is a typical data recording format for a tree plot, which will have a species name, girth, and then height, so on and so forth. And then when it comes to the shrub plot, I told you that we have a nested plot technique where for 20 by 20, for all tree species, we take 20 by 20. We count all the trees that are there in this 20 by 20. But for a shrub, we take 10 by 10 centimeters. A shrub is defined as the one which has less than 10 centimeters girth at breast height. GBH means girth at breast height. DBH means the diameter at breast height. Uh, that means an average breast height of an Indian is 1.3 meters. That's what we follow. Or it should be less than 1.5 meters of height. So we call it as a shrub. Okay. So parameters to be measured in a shrub plot is what are the species that you have, diameter, and then height of the shrub. So you can use a telescoping rod to measure the height. And if its shrub is a leaning shrub, then we have to take the height of that particular from this point. Okay. The summary steps is that for the shrub layer, you make 10 by 10 shrub plot, and then enumeration is done from corner A, and then measure the diameter using a vernier calipers, measure the height, record the data. So this is how you have the data, and then the observations are done. Okay. When it comes to the harvest, we may require to harvest a shrub to look at the dry weight of it, so that the carbon inventory can be now complete. Because if it is a shrub one, this is necessary that we need to take around 2 meter by 2 meter quadrant outside the shrub quadrant, and measure the take entire harvest it and dry it out. And this protocol, incidentally, was developed in association with Indian Institute of Science, CIRSI, when we conducted a series of establishment of 1 hectare plot around CIRSI. And then much of my talk is also, some of the pictures are also derived from the Indian Institute of Science plots. And herb is a one, all those individuals which are less than 1.5 meters at breast height, and then grasses, seedlings, everything is now included as a herb here. And then we only look at the number of ferns and how many species are there, and then what percentage of area is covered by the grass. So we make 1 meter by 1 meter quadrant here, and then split the quadrant into 4 smaller ones, identify the species, count the individuals, and then make it, if it is a grass, you can develop into, if you do not know the grass, you can make into, a temporary species like species 1, species 2, species 3, make a herbarium, go back and identify back. Okay. And then we can make an abundance category if only one particular type is abundant, and if it is occupying more than 75% of the area, call it as abundant. You can call it as frequent, occasional, rare, like that, based on the area that is actually covered. So you should also harvest the herbs if at all you want to make a carbon inventory of the herbs, usually usually away from the herb plot in the next plot of this kind. Okay, these are the formats that we are talking about. And in fact, most important aspect in forest monitoring is to look at the canopy. So what exactly is the canopy? The topmost layer is called as the canopy. How exactly we look at it and why we should look at it is that whenever there is an opening of the canopy, the gap that created due to the canopy opening will be the starting point for most of the recruitments that happen in a forest. This is called gap phase regeneration. As you see, if you walk through a forest, if there is a small gap, you will find dozens of small seedlings regenerating. Hence, estimating the a canopy opening is most important. In fact, most of the disturbance are directly related to the canopy opening. So that's why what we do is we uh, we take up what we call it as a score method, and then entire 1 hectare is divided, as we know that 20 by 20 grid sits there, and then uh, so we can walk along grid line A and then stop at every 20 meters, and then you can take the opening. Like, you have it here. Yeah. So you can use either a densiometer, which is an extremely simple instrument to calculate what percentage of your canopy is covered, or you can even score based on whether it is 80% cover or 50% cover, or like that. So this, I would call it as around 50% of the canopy is covered, other 50% is just open. So you might have, if the coverage is 80%, then you have to just mention that. So we have multiple data points coming from a single 1 hectare plot for the canopy, and then we can take the average of it. And another most important aspect in forest monitoring is the method of estimation of the litter. So litter includes any detached part of a particular plant. It could be a fallen off bark, it could be a small branch, it could be a majority of it is dry leaf, reproductive parts, flowers, fruits, trash, whatever it is. So we put the litter traps like this, 1 meter by 1 meter litter trap with a small plastic sheet or a plastic net attached to it. You can actually put these such litter traps all around 1 hectare, and then start collecting periodically litter and estimate what would be the litter fall through the season. Litter traps are used for collection of the microlitter. Yeah, this is either 1 meter one or half a meter can be now made, and then we can collect 20 traps in 1 hectare. That's quite a large number of sample. And then once you have the litter, you can group it as a leaf litter, flower litter, so on and so forth. We should also have to take the dead and fallen measurements had to be done. What is the kind of dead that we have? You have dead and standing, dead and fallen, dead and decaying. Select every plot there, and then look at the fallen trees, measure the girth, at least in 3 different points along the girth, and then make a format. Okay, so this would actually tell you how exactly the recycling of the entire carbon can happen. So this is in nutshell about how we establish a permanent vegetation plot. Apart from this, as I said, national forest inventories use transect method. Some use cluster circular method, systematically put all through the forested area, and each one of them have their own limitation, and each one of them have their own advantages. But by far, 1 hectare plots have been used in many countries, successfully monitored for more than 3-4 decades. And then we have chain of these 1 hectare plots occurring all through the Western Ghats that are very important for monitoring the vegetation. So thank you very much for your kind attention. Let me thank the organizers for this wonderful opportunity, and also look at this African woman who is the first recipient of Nobel Prize, Wangari Maathai, for her contribution for reforestation. Thank you very much. I can take some questions if there are any.
Thank you so much, Vasu sir. By living in an urban classical setting, we don't know such a beautiful forest is also there, and such parameters are there which can be studied. So thank you very much for your wonderful presentation and very excellent way of explaining. Many people in YouTube live streaming are replying. They are telling that it is a very wonderful presentation they have seen for the first time.
Yeah,
So
thank you
can
very much.
I
Thank
request Ravikant sir, can you start sharing the screen?
Dr. Ahmed, I also invite
I'll do that.
students to Sirsi for the first-hand information about how do we actually the 1 hectare permanent plots look like. So you can visit sometime and have little fun. Thank you very much.
Thank you. Thank you so much, sir. Have a nice day, and thank you. It's time to welcome our well-known, renowned speaker. He's very much busy. Always nowadays, he's found a lot of time giving training programs in Northeast. So I am very fortunate to get Dr. Ravikant. He's a senior fellow and associate professor at Ashoka Trust, that is A3, and he's the senior fellow in biodiversity monitoring and conservation planning. He has research experience of 20 years, and he led multiple research projects focusing on economically important trees and forest genetic resources. He also applies molecular tools in context of tropical forest conservation and species recovery. He has also addressed the impacts of harvesting on the genetic structure of medicinal plant populations. In recent years, he has developed DNA barcodes for a number of medicinal plants and has assessed the extent of adulteration in the medicinal plant trade. A core research focus has been on enhancing the conservation and sustainable use of forest genetic resources and in developing tools and research outputs to manage forest genetic resources of Western Ghats. He has been part of the National Mission on Biodiversity and Human Welfare, which has been funded by the Principal Scientific Advisor, Government of India. As a part of NBMHW, he is coordinating component on biodiversity and agriculture, which aims at ensuring food and nutrition security by incorporating sustainable use of biodiversity. He has published more than 100 research papers in various journals of high peer impact factor, led more than 20 research projects funded by DBT, DST, NMPP, and other international funding agencies. To his credit, research specializations are DNA barcoding, biogeography and phylogenetics, population and conservation genetics, chemical ecology, and bioprospecting. With these words of introduction, I would like to request Ravikant sir to begin his talk. Over to Ravikant sir.
Yeah, thank you very much for that nice introduction. So today's what I'll be talking on is conserving genetic resources of plants. So before this is what I'm going to be talking on. Four major themes. One is trying to give an issue about why we need to conserve genetic diversity, and I'm going to stress on why is it important to conserve genetic diversity. And then second, I will be talking about what are the issues in conserving genetic diversity, whether it could be a plant or animal or any other organism that you're looking for. And how do we measure genetic diversity? I will not talk much about it because you already had some first speaker talking a little bit on that. And then I will talk about examples of how you can use genetic diversity for conservation. So as you know, India is a very mega diversity center, and we have about 4 biodiversity hotspots in the country. And if you look at the diversity, we have 3 levels of biodiversity. When you actually start looking at it, one is ecosystem diversity, where you look at different kinds of ecosystem diversity, whether it could be wetland diversity or whether it could be tropical diversity, tropical forest diversity, or things like that. And then second level of diversity is what we look at the species diversity. You have different species within each group. For example, if you look at plants, you have different families, and within families you have genus. Within genus, you have number of species. So you look at the species diversity. The third within that is the genetic diversity, and genetic diversity is within a species. Okay, it's not across species, but within a species that you look at different populations of species and look at the diversity. So if you look at the visibility, you will see that ecosystem diversity is clearly visible, species diversity is clearly visible, whereas genetic diversity is not very clearly apparently visible. And if you look at the complexity, measuring genetic diversity is complex compared to looking at the ecosystem diversity. But what is genetic diversity? Just to give a brief for those of you who are new to this area, when I talk about genetic diversity, I'll talk about the total number of genetic characteristics that make up a species, or to make up the genetic makeup of a species. Okay, and when I talk about genetic variability, it describes the tendency of these genetic characteristics to vary. And as, for example, as diverse as we may be, all human beings belong to a single species called Homo sapiens, and that is defined by our capacity to interbreed. Though we may look very different, we may look as a you have different, we may have different colors, we may have different heights, we may have different nose lengths and things like that, but all of us belong to a single species. Okay, and then so okay, and but why do we need this diversity? Why do we need diversity? And as you know, genetic diversity or the diversity is the basis for evolution. And as you know, and all things on Earth evolved because of the diversity that existed because them, existed within them, and that is what sustains life. And I'll give you some examples of how diversity is important for sustaining life. But there have been a large number of threats to this biological diversity, and it could be because of extraction pressures, because of land use changes, or deforestation, or extraction of any one particular species at a very large scale. And because of this, you could have a large number of threats, and you could have a lot of impacts on this species at the species level. For example, just to give an example of medicinal plants, if you look at medicinal plants, there are about 800 medicinal plants that are traded in India, and out of that, 61% are obtained only from the wild. And because of excessive harvesting of some of these medicinal plants, many of them are under severe threat, and a number of populations of these species are already facing extinction. So if you look at medicinal plants, there are about 960 medicinal plants that are a source of about 1,289 medicinal raw drugs in the country. And if you look at where they are sourced from, you will find that most of them are sourced from the forest. 52% are sourced from the forest, and only about 20% are actually under cultivation. So large number of species are actually extracted from the wild. And because of this extraction, for example, you have number of species of medicinal plants that are globally threatened. And when you mean globally threatened, or and because many of them are endemic to some parts of the country, for example, this particular species, which is endemic to the Western Ghats, the Myristica species, and their extinction would amount to global extermination of the species. Okay, similarly, for example, Saraka Ashoka, you have for this particular species, the important thing is, like, the medicinally important part is the bark of the tree. And because of harvesting of bark, or because of harvesting of roots of many other medicinal plants, or harvesting of wood or stem or the whole plant, you will find that because of harvesting or harvesting of the bark, for example, could lead to the death of that particular tree. Okay, because if you over harvest. And if you look at the medicinal plant trade, you'll find that about 66 medicinal plants, you either harvest roots or bark or wood or stem or the whole plant. And because of this, we find that a population size of number of species is reduced, regeneration is poor, and because tree species take a long time to grow, and because of harvesting roots or bark or wood leads to slower growth. Similarly, if you find, for many species, you already have incrementally low population size. Okay, for example, Caesium travancoricum, there are now breeding individuals. There are only about 20 individuals, whereas similarly you have very few species or individuals of Persea macrantha or Cosina fenestratum, and these individuals or populations have very few individuals. Like as I told, Caesium travancoricum, you have in a population only about 15 to 20 individuals, breeding individuals in a population, and that is a very, very, very low population size. Okay, and these fragmented populations cannot, with few individuals, will not be able to breed among themselves, and there would be inbreeding leading to I'll come back to what would happen because of inbreeding. And for large number of species also, large number of what we call as non-timber forest products, which are medicinally important or otherwise, we harvest large scale harvesting of fruits. For example, in case of Emblica, the fruits of Emblica, which is harvested quite widely all over the Western Ghats or all over India as such. Large scale harvesting of fruits has severely affected the regeneration of the species, especially in the wild. I'm not talking now, it has come into cultivation, but in the wild, there has been a severe impact on the regeneration of the species. And destructive harvesting has also led to reduced fruit set. For example, you will find that instead of harvesting only the fruits, they often cut the branches to harvest the fruits. And because of this destructive harvesting, the fruit set also has reduced in many individuals of this particular species. So if you look at the threatened status of medicinal plants in India, you will find that almost about 19% are critically endangered, 29% are endangered, and about 39% are vulnerable species. So you have large number of species of medicinal plant which are under threat category. and largely because of over extraction of these species, and largely because many of these populations are fragmented. And consequently, what happens because of extraction, over extraction, you will find that scores of species are rendered rare, endangered, or threatened. There's fragmentation of populations, and there is loss of genetic diversity in these populations and loss of alleles. And because of all these, it could lead to global extinction of species. So why should we bother? Is genetic diversity really important? So I'll just give you an example. I'll just, as you know, when I talk about genetic diversity, as I told, it describes the genetic structure of populations, and it talks about alleles in the population. So I'll give you an example of a cheetah for why to impress upon you people as to why genetic diversity is important. So if you look at cheetah, cheetah went into a genetic bottleneck about 1,000 years back, because of the low population size. Because before it actually got extinct in India, you'll find that there was large-scale hunting of Indian cheetah. In fact, the kings and royals during the early period of 16th and 17th century thought, even the Britishers thought, that it was a trophy hunting. Cheetah was a trophy hunting animal because of its speed and other things. It was considered as one of the greater achievement for a hunter. And because of that, large-scale hunting happened of cheetah in the 16th, 15th, 16th, and 17th century. And because of this, the population size of this cheetah decreased so much that wherever the populations exist, there were very, very few. And because of that, they started interbreeding among themselves. There was close breeding among the remaining individuals. Because of this, what happened? There was low levels of genetic diversity, and heterozygosity is also another way of representing genetic diversity. So the genetic diversity was really low. Because the very low genetic diversity, there was high infant mortality. Because of low genetic diversity, the young ones were not able to cope up with either to different temperatures or to different diseases and pests. And because of this, there was high infant mortality. About 30% of all the offspring that were born died. Similarly, there was also low sperm count in the males, and because of that, the fertilization was very, very poor. And also, the captive populations were also susceptible because they had very low genetic diversity. And because of all these reasons, the cheetah became extinct in India. Similarly, if you look at a bird called Mauritius kestrel, you'll find that because of excessive use of DDT in the agriculture, and because these birds fed on insects which actually were affected by DDT, because of this, the DDT impacted the birds so much that the eggs of these kestrel birds were so thin. The eggshell was so thin that the hatchlings almost failed to survive. Okay, they hatched even before they were actually matured, and because of this, there was a drastic decline in the population of this Mauritius kestrel. And in 1974, there were only 4 birds, and people started doing species recovery of these birds. And what happened was it had 72% lower diversity compared to other species of diversity. And because of this, though they were able to recover this particular species, but still it is under a threat that anytime if there is a disease attack, this particular species may go extinct. And this is another classical study done on butterflies, where they showed where actually they had different populations of butterflies which had different levels of genetic diversity. There were populations which had high diversity, and there were populations which had low diversity. And these populations were actually left out in the field, and then they were recaptured and they tried to see how many of them survived. And what they found was those populations which had very low diversity, their probability of extinction was very, very high. Whereas those which had high diversity, their probability of extinction was very, very low. Also, it's so clearly indicating that genetic diversity is important for survival of a species. Similarly, it's not just true for animals or birds or insects, but it's also true for plant species. So if you recall our green revolution in plants, green revolution in agriculture largely happened because the diversity actually was made to increase. And that happened because they bought in, for example, in case of finger millet, they bought Indaf variety, that is African variety, bred with Indian variety, increased the diversity. And because of that, the crop yield was very, very high. Similarly, for example, if you look at here, genetic diversity is directly linked to the number of seeds per flower stem. Okay, so if the genetic diversity is very high, the number of seeds per flower stem is also very high. Similarly, number of seeds per plant also is very, very high if the molecular variance or if the genetic diversity is very high. Similarly, the seed mass, the seed weight, for example, is also very high only if the genetic diversity in that individual is very, very high. And there have been number of examples with number of plants to show that if the genetic diversity is high, the seed number is high, seed weight is high, and therefore the total yield per plant would be very, very high. And that is what was exploited in the Green Revolution. And not just that, genetic diversity is important also for, and this is basis for natural selection. For example, if you recall, I don't know whether you people will recall this example which was taught in schools about this English peppered moth. That before the Industrial Revolution, the light colored moth was often seen, and because of Industrial Revolution, this particular moth almost became extinct. But because it had genetic variability, you had a black colored moth also existing, and because of that, this species did not go extinct, but this species survived. So if you look at before Industrial Revolution, because of lot of lichens on the tree, this peppered moth was able to survive because insects were hardly able to see it, and only this black colored moth, which is also the same species but different individual, was picked up by the birds. But whereas after the industrial revolutions, when the soot was deposited on the tree and the lichens died, this particular moth became invisible for the birds, and this peppered moth was clearly visible. And so uh, uh, this particular individuals started disappearing in the population. But because the genetic variability existed in this population, where you had individuals which had peppered colored and which had black colored, this particular species continues to exist today. So these numerous examples indicate, whether it's in mammals or birds or insects or now plants, indicates that genetic diversity is important for survival of species, for the reproductive success to happen, and for species to adapt to changing environment. Whether it could be industrial revolution, which is a man-made environment, change environment, or for the changing environment that is happening now because of climate change. And because of and for this, only if the genetic diversity exists within a population, within a species, that one would be able to survive. But what are the issues in conserving genetic diversity? If you look at what are the issues in conserving genetic diversity, you will find that genetic diversity, one of the main things that impacts genetic diversity is the population size. And genetic diversity is very, very sensitive to population size. And anything that disrupts gene flow, or the interaction between populations, can disrupt genetic diversity and can lead to lower genetic diversity. And if population size is, or if there is movement is not there between populations, and if they become isolated, then inbreeding happens. And inbreeding is inevitable in small population, where inbreeding is mating between close relatives. And if close relative breeding happens, then it results in lower resistance to pests and diseases, and it increases homozygosity, or increases genetic homozygosity, and that results in expression of deleterious alleles, and that also results in extinction of population, or leads to lower resistance against pests and diseases. The third thing is about founder effect, which if a isolated population goes to a few individuals start a new population, their diversity is very, very low, and because of that also genetic diversity can be impacted. But that is not very important. These third and fourth are not really important, where the founder effect is when isolated population started by few individuals, and the bottleneck effect is because of some drastic impact, because of, for example, say landscape level changes. For example, an earthquake separates two populations, or wipes away one large chunk of population, and only few individuals survive. Even then, the genetic diversity is also very low. Okay, so that next generation will have very poor diversity compared to what existed earlier, and because of that, also the genetic diversity is there. The last one on that is the genetic drift. Is a random thing, which is a statistical probability of what we call as genetic drift, which is a random change in allele frequency, and this random drift can happen if the population size is very low. If the population sizes are very small, the genetic drift can happen at a much higher scale compared to a population which has, say, about 1,000 individuals. Okay, so for that reason, you need to have higher population size in each population. And as I mentioned, if the gene flow is interrupted, it leads to homogenization, and that could result in losing of genetic variability. So what happens in a small population? So in small population, or a population which is fragmented, there is inbreeding or random genetic drift. And both of these could lead to loss of genetic variability, and this loss of genetic variability could lead to reduction in individual fitness and population adaptability. And both of this could lead to lower reproduction or higher mortality. And further, this could lead to further reduction in population size, and this cycle goes on and on and on. So this is called as the extinction vortex. Okay, and so that's what happens in a small population. So how do we measure genetic diversity? I would not dwell upon it too much, because you already had first speaker speaking about genetic diversity and marker and things like that, though he was talking more about chromosomal markers, but there are a number of markers exist where it could be morphological, biochemical, chromosomal, or genetic. And these markers could help in identifying or could help in evaluating the genetic diversity in a population. In recent years, one of the markers that is quite widely used is called simple sequence repeats, or the microsatellite repeats, which are very well used in for a number of species. And these are used for not just for quantifying genetic variability in plant species, but also for all animal species, or for most other organisms. These microsatellites are used, and because they are highly variable, fast evolving, and codominant, and they're though they're relatively expensive, but they're like give a very good data to actually characterize the genetic variability of any species. And they have been widely used for genetic identity, parentage, or identification of clones and cultivars. They have been used for studying phylogenetic relationship among species, though nowadays there people are using a number of other markers and number of other genetic tools for doing phylogenetic analysis. But nevertheless, people have used SSR markers earlier to do phylogenetic analysis. And for breeding purposes, especially in plants, people have been using SSR markers, and they have been used for doing a number of studies for biodiversity evaluation, for germplasm collections, for genotypic individuals, and for genetic analysis. So I'll just give you a few examples of from India of how you can use genetic markers for conservation. And for example, this is one particular tree called Mapia, or also called as Nothopodatus demoniana, which was harvested quite extensively because of the importance of this particular plant for what we call as camptothecin, which is an anti-cancer metabolite. This particular metabolite is very important anti-cancer drug, and the market size is quite huge. And this particular species belongs to Icacinaceae. And because of the importance of this particular chemical, camptothecin, as an anti-cancer drug, this particular tree was harvested throughout Western Ghats quite substantially. And so this, during the last 2 decades, for example, there has been almost about 40% decline in the population of this particular species. In fact, in the last few years, there has been so much of harvesting of the fruits that the regeneration of this particular species also has been impacted in few populations within Western Ghats. So what we did was to analyze populations that were sampled, that were impacted by harvesting, and those which are kept as control, which were there in the protected areas. So here we looked at populations which are outside the protected area, which are like usually harvested, and those populations which are not harvested. Okay, so we sampled about 8 sites, 4 each as a pair. So one is inside the protected area, one outside the protected area. Okay, so these were the different populations that we sampled, PA and their immediate corresponding non-protected area. So what we found was, for example, if you look at the proportion of adults as coppices are nothing but recently cut stems which are regrowing back. Okay, so if you look at the coppices, you'll find that proportion of adults, you'll find that in the non-protected area, there are large number of about 60% of coppices were found. That means that 60% of these adults were actually cut outside the protected area, whereas inside the protected area, there were very few which were harvested. Similarly, if you look at the proportion of cut stems, they were very, also high in the and similarly, then here in this particular case, we looked at using the microsatellite markers, we looked at the genetic diversity of populations within the protected area and outside the protected area, hoping that the populations within the protected area are quite well preserved, because people generally don't harvest inside the protected area. And then those which are outside the protected area are largely harvested. So if you look at the allelic richness, or it is one form of genetic diversity. And if you look at the allelic richness, we found that allelic diversity was low in the non-protected area compared to the ones which with the populations within the protected area. And this was been shown in many other taxa where which are subjected to harvesting. Similarly, if you look at private alleles, the private alleles are alleles which are there which are unique to a population. So if you look at those those private alleles too, they were found to be very high in the protected area compared to those which are outside the protected area, indicating again that because of harvesting, we have lost number of private alleles. Similarly, if you look at rare alleles, okay, for example, this one set of population which are looked at Talkaveri and Bandikadu. So there are, the protected area within Talkaveri had, for example, almost about 50 rare alleles, whereas just outside the protected area, outside the Talkaveri populations, which are like again subjected to harvesting, they had very less than 10% of the, this is comparing between protected and unprotected area, Talkaveri and Bandikadu. So if you look at the rare alleles, there are about 50 rare alleles in Talkaveri and about 30 in the non-protected area. Similarly, if you look at common alleles, there is not much change. Okay, but the rare alleles are actually lost when their harvesting is small. And there have been a number of studies earlier also which have shown this, where in control populations there seems to be less disturbance compared to less loss of diversity compared to non-protected area compared to protected area. So we did also this particular study in another site and another 3 protected areas. One is in Biligiri Rangan Temple Wildlife Sanctuary called BRT Tiger Reserve. Then in Hills and in Kaveri Wildlife Sanctuary, all in southern Karnataka. And in these 3 sites, again, we looked at a number of other species, including Philanthus emblica, Philanthus indofishery, Boswellia serrata, and Termina cebula. All of them are what are harvested for their fruits or for their, for example, Boswellia is harvested for resin, but the other 3 species are harvested for their fruits. And because of exclusive harvesting, again, there has been a problem with their regeneration, problem with a number of other issues are there. So we looked at a large number of individuals across these protected areas. We looked at adult, we looked at senior, for example, in BR Hills, we sampled so many individuals, and what we found was that the heterozygosity, or what we call as genetic diversity, for example, in these 2 species. So if you look at the control and harvested population, or harvested is one which are outside the protected area or towards the periphery of the forest, where it is largely harvested, we found that those compared to those in the control, which are in the core of the sanctuary, the diversity has actually reduced in the periphery of the sanctuary. So that is what we have seen. And this was also done a long back again in another species called Termina bellarica, where they have seen that because of harvesting, there has been genetic differentiation of populations. That is, the control population had very different genetic diversity compared to those outside this one. And they had done the study in BR Hills and Madhumala Wildlife Sanctuary, where they also showed that control populations was unique because it was not harvested, whereas highly disturbed and mildly disturbed populations were completely having different genetic diversity. In terms of that, they showed that the genetic diversity of those populations were very low. Embryo abortion, in fact, and because of that, we also find that the cebula regeneration also is very, very poor in all these 3 protected areas. So can we identify thresholds of harvesting from these studies? Can we regulate levels of harvesting so that we avoid genetic differentiation or avoid genetic loss of populations? And what should be the long-term interest in improving or conserving genetic resources of such species? So that is what we have been working on in the past few years, where we are trying to develop sustainable harvesting techniques, and then developing, say, something called as decentralized enterprises in these 3 protected areas, where we are setting up enterprises to increase the income of NTFPs, developing capacity of people there, and trying to tell them, that instead of harvesting all the fruits in a tree, harvest only about 70% of the fruits and leave 30% for regeneration to occur. Harvest only the low-hanging fruits, leave the fruits on the top for the birds to disperse, and then also, but at the same time, we're trying to see that their income is not hampered, that their income is still there. By trying to see what we have done is we established enterprises to actually do what we call as processing of the harvesting. Instead of directly sending the fruits, for example, in this case, amla or the philanthus fruits, instead of directly selling it, trying to teach them to make pickles, trying to make candies out of that amla, and then get more income from even less harvest that they do. So that is what, so we are actually providing value addition to many of these species, and by thereby increasing their income as well as trying to see that the harvesting in the forest is actually reduced. So they have also created this brand, and now people are doing in these 3 sites. People are selling a number of NTFP products using this brand. So take-home lessons: genetic diversity is basic for viable population, though it's difficult to quantify, but it's necessary to evaluate critical questions related to conservation of genetic resources of medicinal plants, especially medicinal plants because they're harvested quite widely. And and these genetic diversity can be used to address many issues, especially for genetic enrichment of rare, endangered, and threatened species, for genetic monitoring of reintroduced species, and for undertaking polygenetic relationship among species. Thank you. And if there are any questions, I will take it now. Hello. Yeah. Hello. Yeah, any questions from any participant? Hello.
Sir, thank you very much, sir. I was
Yeah.
just
Okay. Yeah. Sorry. I thought I lost the video. I lost the audio.
Is there any difficulty from our side or
No, no, fine, fine. I don't know whether they were able to hear.
okay? Thank you very much, sir. Sir, can you? I mean to say it is an online platform, but I request from Adri some more people. Can you share the contacts on the WhatsApp?
Yeah, yeah, sure, sure. I will do that. Yeah.
How is Saudra madam?
Ah, she's doing fine. Yeah, she's doing fine. Yeah, I will share the contacts with you.
Okay. Thank you so much. Thank
Yeah,
you so much, sir. Wonderful talk. Such
yeah.
a eye-opening, so great talk, sir. Thank you so much. Thank you. Now
Okay.
I request Chandrakanta madam to start sharing her screen. Chandrakanta madam. Yeah. Let me give an introduction to you, ma'am. It is a proud privilege. This is the second time we are going to listen Chandrakanta madam. Chandrakanta madam is PhD in post-harvest pathology and technology at Eastern University, Sri Lanka, and the Cranfield University, United Kingdom, as Commonwealth Scholar of the United Kingdom. She has been serving as senior lecturer for last 20 years at the Eastern University, Sri Lanka. Her research findings have been published in several national and international reputed journals, as well as in many national international conferences and symposia, including United States of America, Australia, New Zealand, Turkey, Dubai, China, Mauritius, Singapore, India, Sri Lanka. She became one of the nominees among the first three, in fact, the first ever female nominee for the Vice Chancellors of Eastern University, Sri Lanka in 2018, to be appointed by His Excellency, the President of Sri Lanka. Throughout her career, she has been engaging several administrative positions at university as head department of botany, director, Center for Information Communication Technology, director, Center for External Degrees and Extension Courses, director, Career Guidance Unit. In addition, at present, she is holding the following positions at the university. President of President of Sri Lanka University Sports Association, which is affiliated to International University Sports Federation and Asian University Sports Association. Vice President of the Federation of University Teachers Association Sri Lanka. In addition to that, due to her interest in social service, she became a member of International Lions Club, in which she was a career charter president at campus Lions Club of Eastern University Sri Lanka in 2010, and currently engaging a regional consultant for the Eastern Province Sri Lanka in the service organization. With these few words of introduction, I would request ma'am to begin her presentation.
Hello, good afternoon. Thank you so much, sir, for your wonderful introduction. Very soon, I think I have joined back with you. So thank you again for inviting me and remembering me, of course, to have this lecture. Sir, can I have just a question before I start my lecture, whether that participants are themselves muted or you have muted them? Because, I mean, in between, they are not allowed to ask any questions, as I think. Am I right?
Not allowed questions is because of time scarcity. We are not making
Right.
this, and the program is also live on YouTube. If some questions
Okay,
are seen, you'll be passing it out.
right. Okay, that's fine. That's great, because this is being a lecture. I thought that whether they are allowed to ask any questions. Okay. Okay, because of your time limitations. Thank you so much, sir. Let me continue then. Can you see my slides, please?
Yes, your slides are visible.
Okay, fine. Thank you so much again, and very good afternoon to you all from Sri Lanka. And we are fortunate that we are being neighboring countries. We are in the same time, so it's good afternoon now. Well, again, I'm going to have my presentation. This is actually a lecture. Just last month, I had my guest lecture in a conference with a user. Thank you again for this opportunity. But this is completely a different topic that I'm going to talk. As in my introduction said, I'm a senior lecturer in botany. Of course, I'm working in, my major is actually the post-harvest disease and technology, but at the same time now I have been engaging in some research work in the harvest, that is post-harvest products, as the medicinal plants, leafy vegetables, because I just wanted to study some traditional, get back to our traditional medicinal system, just because of the alert that we got in during the COVID-19 period. And again, continuously with sort of new pathogens, new viruses, new bacteria, which we are struggling. Even the medical field is struggling to control the diseases. Still more than a year now, we have been in COVID-19 pandemic situation, but still very recently we have come across with the vaccination. So until such time, very unfortunate that we have lost so much of human beings worldwide. So this is why I have just thought to switch into and expand my research area, like some more which we can correlate with our traditional medicinal system actually, which have been there for years and years, for ages it was there. But very unfortunate that with the recent development of technology, that and this, we have almost completely, most of us, particularly our youngsters completely have forgotten about our traditional medicinal system, which are so safe and which are so absolutely health environmentally friendly one. So this is where I just thought to get back to that system through undertaking some research work about these naturally available medicinal plants and their medicinal properties, and what the reasons behind that for these medicinal properties. So on this basis, I'm going to talk about the some of the antibacterial activities and some of their properties, actually the phytochemical properties of some medicinal plants which have been so far studied. But of course, I'm not going to cover all the plants what we have studied, but I will be just looking at some of the plants which I have so far studied, our students have studied and found their properties, as particularly the antibacterial or antimicrobial properties. Again, move to my introduction. Yes, as you all know, I think you being mostly this lecture is for the postgraduate, this is MSc students. I'm not that sure, I mean, your major actually, but I'm sure that you all are working with sort of botany area. So this is totally regarding our plants. Maybe it is medicinal plant, but this is the medicinal plants which have been considered as medicinal plants, but those are weeds. Most of them are weeds in our environment. Again, I should say that like in India, here in Sri Lanka, because we being the neighboring countries, we are blessed. We are our geographical area, our geographical pattern is we are blessed totally with the natural available plant diversity, which are having so much of medicinal values, so much of phytochemicals are hidden inside it. So it's our duty, as we all know, it's our duty now to find it out, to do the expand our research work to find their medicinal properties completely, and to get back to our traditional medicinal system to protect us from all such existing or newly coming all these sort of dangerous pathogens actually around us, which we have been facing very recently in a crisis situation. So that, as I said, the plants have not only the nutritional values, we don't just eat them just for the nutritional values, but they also having the medicinal, ritual and magical values, of course, as per some of the few researchers have told in 2014. It has some magical values which are very, very, environmentally friendly one and health friendly one. So the green plants, importantly, the medicinal plants are a rich source of nutrition as well as containing medicinal value too, again, as per one of the Indian scientists in 2013. The medicinal properties of plants may be based on the antioxidant and antimicrobial effects of the phytochemicals, as per, again, another Indian scientist stated in 2015. So it shows that the Indian scientists have done a lot in finding their nutritional values as well as their phytochemical values, also importantly, their antimicrobial values. So the plants, let me move to the introduction a bit fast. The plants are producing a diverse range of bioactive molecules, so making them rich source of different types of medicine. This is again and also just quite a recent study done by, again, an Indian scientist. So it could be noted that adequate number of studies have not been done so far. This is the concern of the scientists now, particularly the botanists now, because the biodiversity in India, it can be biodiversity in Sri Lanka. It's still we did not reach what we are supposed to reach actually through our research to find the medicinal properties or the medicinal values, their nutritional values of this biodiversity of these medicinal plants or whatever of course the medicinal plants what we choose from the environment is can be any other plants or the plant plants. So those are now each and every weed if you find in the environment. Each and every plant, each and every weed, what we just throw, we just ignore in our environment, having these medicinal values, what the scientists have been now recording throughout in the recent past, particularly about their phytochemical values as well as their antimicrobial values. As long as if you take the country Sri Lanka, let me just touch something about our Sri Lankan situation about this herbal traditional medicinal system years, rich historical background of practicing this traditional medicinal system totally depend on these plants, actually plant diversity. Currently, it can be found that 4 traditional medicinal systems in Sri Lanka, namely Ayurveda, Siddha, Unani, and Desha. These are some sort of the language from other one of other major languages that in Sri Lanka, which is Sinhalese names, actually almost similar names in our language as well. It's very common. So these are the 4 different types of traditional medicinal systems being practiced in our country for more than actually, as I said, 3,000 years. So this is the pictures. Of course, you can see the pictures. This is how the Ayurvedic traditional system, the people are, they're recognized or they are identified by their, external appearance actually. So immediately we can just medicinal expertise of course in that field. So the present reports indicated the availability of 1,430 species which are belongs to 838 genera and 181 families of medicinal plants in Sri Lanka. Out of them, 174 species, nearly 12%, are endemic to the island. That is very fortunate. We are so lucky that. So the nearly 12% of these existing medicinal plants, so which are be used as the medicinal plants among the biodiversity of plants, each actually is endemic, what the researchers have mentioned so far. Also, 150 medicinal plants are commonly used in traditional medicine, while 50 species are heavily used. Very, very recent studies have reported this. Nearly 50 species are very, very heavily used in recent past, because now the people are getting the concern of this traditional medicinal system. It's not just because of they lost the faith in the English medicine or the Western medicine. It's not just because of that, but what we see right now for the last 1 year, nearly more than a year, but we have seen actually so far is, if there is a sudden crisis, if there is a sudden problem, health issues occur, or if there is a sudden pathogen, it can be sudden bacteria or the virus or whatever is newly emerging, we don't have an answer in Western medicine. Absolutely, we don't have an answer. Where unfortunately we have lost millions of human beings, and by the time we come across with some solutions for this problem. But where, if you look at our history, even in Indian YouTubes, you can simply found that there are enough YouTubes have been published in the YouTube actually, which are actually by the very, very old, senior people, saying the medicinal properties of these plants is really surprising. If you refer all these YouTubes or the records in the internet actually, where they have simply lots of reports have been accumulated about all such against all the traditional medicinal system or the traditional medicinal plants or the plants, the biodiversity, the number of plants which we can simply use against any sort of health issues, in any sort of health issues. Even is a very good example against the coronavirus. There are a number of plants have been already they have reported. It's not just coronavirus actually, it's the virus where we don't have a medicine, we don't have a vaccination. So in that case, they have already been published. They have number of plants which can show simply, which can increase our immunity naturally in our body just by having a 1 or 2 days just boiling the leaves or something and drinking, just simply it increase the immunity in our body against the viral diseases. That is very important. So why I'm stressing here as viral diseases, just because we don't have an answer in our Western medicine for these viral infections. If there is another new virus tomorrow, there is no answer at all from the Western medicine. Whereas this traditional medicinal system, these medicinal plants or the weeds which we are now labeling as the medicinal plants, actually they have the answer against any sort of infections, particularly any sort of viral infections. It can be chikungunya, it can be corona, it can be any other viral diseases, viral infections. There are a number of medicinal plants already available for us, which is without any problem, any side effects, and there which are increasing what we so called that immunity, increasing the immunity. So there are plants which are giving an absolute answer for us against all such health issues. This is where the traditional medicinal system is now little by little is being very famous among the public or among the health sectors, even in our country, even in India or many other countries. So despite the influence of Western medicine, the use of traditional medicine is increasing day by day, almost in every country, not in the Asian countries, almost in every country. So due to the abundant availability of economically favorability, brings Sri Lankan people to rely on this traditional plant medicinal system. However, very unfortunate, very unfortunate, the threats towards these medicinal plants of Sri Lanka. Why? Not even in Sri Lanka. This is common in everywhere also. The countries which have all these plant diversities as the medicinal plants or green leaf vegetables or the weeds or so anything. So there is a threat for these plants. How these threats? Why these threats actually? See how the situation in Sri Lanka. About 80 medicinal plant species are now known to be at risk. Not just 1 or 2, 80 different species of medicinal plants. The medicinal plants population in Sri Lanka is adversely affected by this is because of over-harvesting, because the people are now get attracted towards this traditional medicinal system where the over-harvesting is there, but we are not replacing the environment what we harvest there. That is another totally a foolishness what we do. We just concentrate on harvesting, but we don't think that, we don't look into that, give it back to the environment. That is replanting these medicinal plants or having the medicinal plant gardens or whatever. And poor harvesting practices also from the wild. Just we harvest and come. Moreover, unsuitable agricultural practices, another reason why the threat is there. Why they are becoming extinct, why these medicinal plants, or why these the plants which are having the medicinal properties, why they becoming extinct so far is just because of some of the agricultural practices where the shifting cultivation chain, cultivation, all these things where it is get destroyed, burning the lands, and even the urbanization where the plant diversity is being lost day by day. So this is where the threat towards not just towards the medicinal plants, all of the country, I would say, the threat to the plants, our biodiversity of the plants. So as well, pollution, urbanization, all these things, the things leading towards the threat of the extent of the several blessed medicinal plants and other plants. However, through the World Bank funded Sri Lankan Conservation and Sustainable Use of Medicinal Plants project, there is a project which is funded by the World Bank. It was established the 5 of medicinal plant conservation areas, which is a very big areas, which is under a project undergoing in Sri Lanka, which is a huge project just to conserve the medicinal plants, or just to conserve the biodiversity of these plants. The established area in some of the very common areas in Sri Lanka, that is named here, where this establishing the medicinal plant gardens is under the project is carried out. The villages in and around these conservation areas have been trained to harvest the medicinal plants sustainably. This is where everywhere we use the terminology, the sustainable use. So wherever the medicinal plants, the traditional medicine are being mostly practiced by the people in rural areas, not in the urban areas. So where these people have to be educated, to harvest these plants in a sustainable manner and to conserve this plant diversity. Also, the practices are also being given to the local to cultivate the medicinal plants. So this is what the project mostly concern, and to cultivate to replace these plants to the environment and also to use them in a sustainable way. So So let me give some examples, which is very, very common in Sri Lanka. I'm so sure this, as per the geographical, almost close geographical pattern of Sri Lanka and India, you also will be having, or you, instead of saying having, you also will be blessed with all sort of these plants. But just look at this. This is just a weed. I just want to show you some examples how much these plants are treated and how these we are blessed to have these plants with such a very good medicinal properties. It's absolutely a weed, but you see here is just totally it's a weed. It's a weed everywhere. In the wild, in the forest area, everywhere, it's just a weed. You can see the name and everything I have given here below the picture, all the details. So which is having a very, very good medicinal values, just simply kind of an arthritis, which is having a very good solution for arthritis problem. So which is actually is done by a scientist in botanist in 2030. Again, he is an Indian scientist. So likewise, some of the examples I can show here, which are mostly these are some wild, either they are wild plants or they are just the weeds, which are having very excellent medicinal values. And it has been just proved by the very recent research, not, I mean, earlier researchers, very recent researchers also, again and repeatedly, they are reporting what are the medicinal values these weeds or the plants or the wild plants are having. If you look at here, it's a wild plant, which is having an anti-inflammatory facts actually, which is absolutely a wild plant. And if you look at here, another shrub, which is again a wild. So simply this is treated for the problems related to the urinary tract and all. So this is another one. Again, it's another wild plant, which is again has been shown that very recent, the research in 2013 has shown see that it has the antioxidant, antimicrobial, antidiabetic, and so much of medicinal properties. It's just a wild plant. And if you look at here, some of the other shrubs, which is very, very common, very, very famous for the diabetes. Very, very famous. This has been again proved in one of the researchers by 2019, very, very recently, which has been effect against this diabetics. So if you look at most of them, here this is another very, very common weed, and it's a wild again shrub, which you can see how much of that the medicinal values it possesses actually. Each and every part of this plant, each and every part, I just don't want to mention the names, it is given here very clearly. So here, the thing is, each and every part of this plant is being used as a medicinal property here. Again, so much of, very, very common, of course, for the diabetic, very much for the diabetic, but at the same time, anti-inflammatory, antioxidant, and so much of medicinal values are behind that. So if you look at here, is another endemic plant to Sri Lanka, which is locally named as it's a tree again, which is having a very good anti-arthritis. It is. So you can see here using, this is, very common in India also, and very, very famous medicinal plant actually, which is very commonly used for the diabetic problem. And here now the syrups are available. They're called Nelly syrups. It's available in bottles in very local markets. And not just it is available. It's a very high competition. Even the most of the patients, diabetic patients, are started to use this syrup actually, because it has been proved that diabetic problem. And if you look at here, other which is a wild plant. So most of these are wild plant which are having very good medicinal values, and most of them have been now introduced as in the market actually. Just the fresh flowers or the leaves or all these things are just dried and get packeted, where it is being just as drink, as a herbal drink. That's it. Nothing else. Just is boiling and drinking as a herbal drink, maybe every day, or depends on the patients actually, which is actually prescribed by these traditional people actually who are engaged in the traditional medicinal system. So these are, again, if you look at the leucas, is a very, very common weed. You all must be knowing, being the botanist post-PhD students, you all must be knowing it's a very common weed. But again, it is having a very, very good effect, particularly in Sri Lanka for the children. This is being used because it's a very good anti-worm property it is having, the drink of this weed. Again, this is, you can see here, which is available in commercially also available. These plant materials are, the seeds and leaves are available commercially also, just to have as a herbal drink against the anti-inflammation and for many problems, even the rheumatic problems. You can see again, which is very, the pomegranate very common, and even this is very being is used, though it is not prescribed but any kind of a medicinal system. Now this is very commonly used in dengue, against the dengue fever in Sri Lanka. This pomegranate is it has shown a very good effect in increasing the platelets have been affected by the suffering from dengue fever. It has been very common in Sri Lanka, I think maybe in India as well. But it is not available these days because of the off season, but very unfortunate. I mean, again, this the production of these pomegranate naturally it is becoming very low, but now people are very concerned after the problem of the dengue. So these are some of the weeds and some of the, this is very common, the name actually. So I just want to show how much we are blessed, with this biodiversity of these plants, which is having true medicinal values and which we have been ignoring for many years and years, not like our ancestors, not like our senior people. We have the youth now actually. We have been completely, we have forgotten about the values of this plant diversity. But it is the high time. It is the high time to get back into to look into our biodiversity of these plants and to conserve these plants and to use this biodiversity of these medicine, particularly the medicinal plants or all the plants, to conserve them, to use them sustainably. Because now it is the time, the alarm is there given already, where we don't have the answer for newly emerging problem, health issues in Western medicine. We don't have any answers from Western medicine. So it's high time for us to get back, to look into this again, the biodiversity, and get back into the traditional medicinal system, where we are, being the Asians, being the Sri Lankans, being the Indians, we are really blessed with all sort of this biodiversity, but we have been ignoring. Where in Western countries, though they don't have all these natural biodiversity there, but they are turning towards this traditional medicinal system, though it is not available. But we are having throughout the year, fortunately, we have that climatic condition, that weather condition, that geographical pattern. But throughout the year, we are being blessed. But we have been ignoring so far. But this is the right time and high time to look into this, our traditional medicinal system and the medicinal plants which are around us, particularly to conserve and to use them sustainably. Again, some of the, more other weeds, this is an absolutely wild plant, but it's a very common in the home garden as well. But it has a very good medicinal property, because this is the leaf juice or the leaf drink is used as an anti-diarrhea one, particularly for the children, which is a safer as well as without any side effects, and then immediate answer as well. Then this is very common, the citrus family, any kind of plants from citrus family. So there are many examples I have given here, which is, I just want to show how much we are fortunate to have this biodiversity of these medicinal plants. Here, this is very common, being the biology students or the botanical students. I think you must be well aware about these plants and the plant parts of this very commonly available plant in India as well as in Sri Lanka. This has now become very famous against coronavirus, the COVID-19. Just as the herbal drink, you can view many sort of YouTubes and Indian YouTubes actually, which are within the traditional medicinal system. This has been long back introduced by those people, the people in the traditional medicinal system, the senior people. Long back that has been introduced against the viral infection, any kind of viral infection for which we don't have answer from the Western medicine. So long back that has been introduced, but we have been ignoring. Now we have again and again now look into this. You can see the video clips, how much they have been viewed actually, because it is giving a promising answer against the coronavirus also. Though it is scientifically, I don't know whether it is clinically reported or clinically proved or not, but it is giving a promising answer. Even as I said earlier, the pomegranate against the chikungunya or against the dengue fever. And likewise here, this plant is giving a promising answer against any kind of viral infection, not just corona, any kind of viral infection, even just common cold. A herbal drink, one day herbal drink is giving a full relief and giving a promising answer. So this is I'm showing some of the scientific methods what we have been using to extract this plant or the leaf or plant part extracts and to study their phytochemical properties. These are some of the common methodologies which you must be aware of that. I just want to show, and this is how we have been, my students have been reporting their properties, the phytochemical properties, and it's very common how these phytochemical properties are get recorded and what are the tests available. And again, how to study the antibacterial activity. There are many methods available. More very commonly, the well diffusion method is being used to study their antibacterial activity. Mostly these antibacterial activities are tested against the human pathogenic bacteria. There are plant pathogenic bacteria also being studied, the antibacterial activities, but very commonly the scientists or the botanists are using the the human pathogenic bacteria, because we are now in the process of finding solutions for the newly emerging, finding solutions for the traditional medicinal system to find the answer against the newly emerging pathogen, particularly the human pathogens. This is where people are looking, particularly testing the antibacterial activities against the human pathogenic bacteria or the fungal or other microorganisms. So these are the methods and the methodologies that how these things are being used. I'm so sure that you being the postgraduate students, I have been there in Maharashtra so many years, both for last 14 years, and I have been visited to many microbiology laboratory. I'm so glad to say that I'm so pleased to say that you people, the postgraduate students, are having very good talents and skills and knowledge. I'm so proud to mention that here. I just want to record that here. You all are very talented, very skillful, and very knowledgeable in studying these microorganisms, their antibacterial activities, or any other research works related to the microbiology. I know that I'm one of the lifelong member in Microbiology Society of India also. So I know how much you all are very much skilled and enthusiastic more than that. I'm so glad to say that you all are so enthusiastic. Study all these microorganisms which are causing problems in human beings. So this is where I don't need to explain too much about the methodology, since you all are very talented in that. So finally, the statistical analysis being done. So this is some of our research work that what I'm showing here, the leaves that we have studied. I think many leaves we have so far studied. So some of the results I am giving here. So this is the well diffusion method and checking the antibacterial activity. So this is how we just get the diameter of the colony and etc. You all are well aware of these methods and methodologies. So finally, before concluding, I just want to say some more few things about Sri Lankan situation in the traditional medicinal system using this medicinal plant. So the Sri Lankan government recently paid very much, it's very fortunate to say that, much attention to the development of medicinal system, that is the conserving the medicinal plants, particularly in Ayurveda, which is a very common medicinal system in Sri Lanka. However, gaps are there to fulfill regarding the conservation of medicinal plants, along with the restricting the clearing of the wildlands. So that is another unfortunate situation. Nevertheless, there is an immediate necessity to comprehensive the pharmacological and phytochemical investigations of traditional formulations of these medicinal plants to get to know their hidden secret on curing these diseases, particularly the newly emerging diseases. In that is very especially against the viral diseases. So this might help the nation to enhance its medicinal system and maybe to get foreign exchange through drugs interventions also. And economically also there are several advantages. So this is what by saying I just want to conclude. So I would like to request you all, being the postgraduate students, MSc students in botany, think of this. How to conserve our biodiversity of plants, particularly the plants which have been recorded as they possess the medicinal values. So conserve it. Make the people, give the awareness to the people to use them sustainably, because we are the people, again and again, I'm telling the Asian people, being the Indians, being the Sri Lankans, we are the people being blessed with all such of biodiversity of this very good, medicinal plant system. So conserve it. being the postgraduate student, do the research to find out the different plants in the plant diversity which are having very good medicinal values, and educate the people to use them sustainably. Of course, people have started the usage has been increased dramatically, but the question is whether those are sustainably used, because those are, again, I'm telling, mostly they are used by the rural people. So give them an idea, give them awareness how to use them sustainably, how to replace that to the environment once it get harvested, how to conserve them. So do research on that. Give the awareness to the government in the conservation. When I was a couple of years back in India, in Maharashtra, one of the universities, I forgot the name, I saw that 100 acres of land waiting this medicine plant. I'm so glad to say that people are very much keen. I know in Indian scientists, Indian botanists, particularly in Maharashtra, they are very much concerned about this conserving these medicinal plants. But this is the message I just want to give you. Being the medicine, being the botanist students, the postgraduate students, give more attention toward this biodiversity of these medicinal plants, particularly conserving and using its sustainability. So thank you very much, sir, again for giving me this opportunity. This is my message from Sri Lanka for you all being the postgraduate students. So I am sure that you will carry my message to others as well. So thank you so much. Stay safe. Thank you again. See you soon physically. Thank you, sir.
Thank you very much, ma'am, for such a wonderful talk. So today's talks come to an end here. So we had four speakers, as you have seen today. The first speaker was Professor Altaf Hussain Nadaf from Savitribai Phule University, Pune. And second speaker was from University of Agricultural Science, Dharwad, Sirsi campus, Professor R. Vasudeva. Then we had from A-Tree, Ashoka Trust for Research, A-Tree, Dr. G. Ravikant. And just a while before, we were participating in the lecture by Chandrakanta ma'am from Eastern University, Lanka. So I express my deep sense of gratitude towards all the speakers of the day who have made our day by delivering their own lectures for our students and our participants. So a big thank you for one and all for joining with us. And I request all the participants to attend again, get back again for tomorrow's lecture. Tomorrow we are having interesting lectures from Center for Dryland Agriculture and University of Mysore. So we are expecting talks on tomorrow of drought stress and climate change, and then bioinformatics and its applications. So with a great ray of hope, I expect that today you have learned so many new things from our experts. So, and we'll continue tomorrow again back. Thank you very much, one and all. I will end the meeting over here. Thank you very much. Thank you, ma'am.